microalgae as well as macroalgae. So that's only we are also considering this particular field. This nowadays, all of them are well known that global demand, the energy only the global demand. Every day we are also running the prices, petrol or diesel. Now only they reduce the prices. The fossil fuel is a question mark. So another 10 years or 20 years after that, all of them are only used for the biodiesel. Okay, next one. So here, the government around the world to current policies to the world will need almost 60% more energy in 2030. So more than 45% will accounted in China and India together. Bioenergy can be produced from different species to macroalgae and microalgae. So most of the work is doing in biodiesel from microalgae. But I am also taking the challenge. One of the students, biodiesel from marine algae is a very, very excellent result. We don't blend with any other diesel. Only taking from the algae, biodiesel from algae. After that, we compare with all the normal biodiesel. After that, running the generator. So that's a very good, lot of challenges, a very good opportunity to our seaweed. So all the members already well known, the Laminaria, alba, Nanochloropsis, Chlorella vulgaris, some diatoms also aquatic biomass, the third generation biomass. Already you know the first generation and third generation. So mainly we are also talking about the enzymatic hydrolysis in the pathway conversion complex polysaccharides. Mainly that you know the fatty acid propel, that is very, very important. Next one, please. So the global problems, already all the members well known, the peak oil future. Now with the Ukraine war, immediately announced diesel, petrol prices also increased. Current oil and diesel prices are in the rise. Climate change emission is another one of the issue. Energy security. And this, increase of food prices. Everything based on that energy sources. Okay. So first generation, all the were mainly used for corn and vegetables and ethanol chemical sugar mainly used for corn esterification process. So now the government also don't take from that corn and other sugar material, you don't take from the oil. The second generation lignocellulose or ethanol enzymatic bioconversion vegetable oil, that's also you don't go for that second generation of the biofuels. The third generation biofuels mainly energy cross bioalcohol, alkyl hydrogen, alcohol oil or biodiesel. So that's only both generation. Now only the carbon negative energy crops, another generation. Okay. So here, the microalgae already told. So last time also, all the member mainly talking about more than 6,500 square kilometers in the coastal area. But microalgae culture very minimum place enough. Immediately large scale cultivation within 15 days. Or Chlorella, Nanochloropsis, Nitschia, any species you are also culture. And we are also going for that lipid content could be adjusted through the changing growth medium composition. You also give some stress. At the time, you produce a lot of fatty acid profile. And they could be harvested more than once in a year. Salty or wastewater could be used because a lot of coastal area available. Within a short period, we are also going for that microalgal culture. And it is non-toxic and mainly that algal lipid. Because all the plants absorb all the fatty acid, carbon dioxide sources. That's only we are also taking from that fatty acid propel, screening the fatty acid. After that, you won't go for that biodiesel production. Okay, next one. So, already mentioned the Nanochloropsis, Aculata, Chlorella, Odoconia, Spirogera, Chlorella, a lot of microalgae done in that other foreign countries also. But in this case, in the seaweed, next one. Next one. So here the microalgae oil content, how much just you are also analyzed. At the time more in Nitschia also diatom is more than 45 to 47 and Potricoccus 25 to 75. So here isocrisis 25 to 33. That's only we also analyze the oil content or fatty acid profile in that particular microalgae as well as macroalgae. Okay, next one. So here comparison of some sources of biodiesel. Naturally that corn, soybean, canola, jatropha, coconut oil or oil for microalgae, macroalgae. Just imagine the microalgae and macroalgae also having large oil yield also very high. But other plants, jatropha you take it, more than 5 years wait. After that only taking the seeds. So last area, you want last area. But you also culture in seaweed and microalgae, very minimum amount of place is enough, and every year harvested, continuous harvest. Okay, next one. So here in our lab, we also cultured a lot of microalgae culture going on in that very nicely. Next one. So within 15 days, we also cultured in the more than 10 tons microalgae culture harvested. Okay, next one. So some of the species mainly microalgae and macroalgae. Okay, next one. So here we are also cultured naturally. This is one of my students, Manivannan. He only completed a very good job in biodiesel from seaweed. Very excellent the product. We are also taking from comparison of green seaweed, red seaweed and brown seaweed also. We collected from Kanyakumari coast. Okay, next. So we are also going for Enteromorpha, alpha, fasciata and Caulerpa, some of the green algae and red algae. Okay, next. So brown seaweed, already you know the Tectiota, Dichotoma, Sargassum. We are also screening all the seaweeds, fatty acid profile. After that you select the particular seaweed. Okay, next one. Okay, some red seaweeds. Okay. Already all of them were well known. Okay, right. Next one. So seaweed cultivation. Suppose you are also our aim, don't exploit natural vegetation. That's only all that the government, National Fisheries Development Board and CSMCRI, all our center also involving a lot of that alternate livelihood. Seaweed cultivation going in coastal people. Now the people also earning more than minimum 15,000 rupees earning only seaweed culture. So that's only we are also cultivated. After that take it from that the seaweed also. Next one. So already trained that particular technology transferred to our Parangipete, Kadalur coastal district and other places. Okay, next one. So already all the members well known. Okay, next one. Okay, next one. Our Professor Kadiresan sir inaugurated this particular program. Okay, next one. So initially already all of them were well known. This is 60 kg. After that minimum you harvested 300 kg you harvested from our nearby mangrove area. Within 30 days. Other places 45 days. But here only within 30 days you harvest more than 300 kg. Okay, next one. Very luxuriant growth in that Kappa pecus. And we are also involving other species also. Okay, next one. So what is the methodology? Very simple methodology. Algal collection and low production cost, high pre-treatment cost, algal strain, moderate production cost and moderate pre-treatment test. This is a simple methodology. Okay, next one. So what is the methodology for seaweed collection? Already all of them are well known. And evaporation technology, the oil was evaporated vacuum to release methanol and a chloroform solution using a rotary evaporator. Okay, next I explain that methodology. The small scale biodiesel also we are also ready to prepare. Okay, next one. So mixing the catalyst and methanol, that is very, very important. 1 gram of sodium hydroxide was mixed with 100 ml of methanol and stirred properly for 20 minutes, enough. And biodiesel production, there's one of the very, very important thing, trans-acidification. There's another one process. The conical flask containing solution was taken for 3 hours by electric shaker at 300 RPM. Oil with the sodium hydroxide methanol solution. After that, settling. After shaking, the solution was kept for 16 hours. That is very important. To settle the biodiesel and sediment layers clearly. Entirely 2 different layers. I will show the photo also. Next one. So separation of biodiesel. The biodiesel was separated from sedimentation, oil by separation, funnel, flask separator carefully and quantified. To sediments such as glycerin, pigments and was measured. Other pipe product also, you are also taking from that product. Washing is one of the methodology. At least 4 or 5 times, well washed with this particular solution. After that only that very clear oil also you are also taking. After that process, drying process, the biodiesel was dried by using a dryer and finally kept under the running fan for 12 hours. And storage, the biodiesel production was measured by using the measuring cylinder. pH was measured and stored for analysis. Further, you are also stored. Okay, next one. So what is the analysis of biodiesel? That is very, very important. It's not an easy job. We are also combined with chemical engineering, our biotechnologists and biology member are also involved. At the time only you are also produce a large scale biodiesel production. So oil extraction, one of the method, trans-acidification process, optimization catalyst, optimization of temperature, time, stirring technology, separation of biodiesel and washing of biodiesel. That is mainly for trans-acidification, ester, separate from the ester. And chemical properties, fatty acid propel, already told, the fame. How much are present in that fatty acid propel? The carbon sources mainly present. And estimation of CHNS, carbon, hydrogen, nitrogen, sulfur. Already that instrument available, immediately analyzed. Physical property of biodiesel, Indian standard method, US 1448. You also analyze all the parameters, normal diesel as well as we are also taking from the seaweed biodiesel. You also check density, how much kinematic viscosity, fire point, and cloud point, sulfur content, cross calorific value, blast point. These are very, very important for chemical parameters, also very important. After that, you want to go for that combustion and performance of biodiesel using the diesel engine. Okay, next one. So very simple methodology. First one, trans-acidification process, oil extraction, oxidate apparatus. That's only we are also minimum level started with this particular chloroform and methanol program using chloroform and methanol. Next one. So optimization of, that's a second important one, optimization sodium hydroxide. So alkaline catalyst sodium hydroxide was used at different concentration to optimize the oil extraction, oil extract. We also give some stress also given. Okay, next one. So two layers immediately you are also separate. One is the topmost methyl ester and another one cholesterol. So the another byproduct also we also take it from this particular biodiesel. Okay, next one. So water washing, already told, 3 or 4 times well washed with this particular, at the time only excess methanol and soap also removed from that biodiesel. Okay, next one. So settling of the residue, the remaining residue was allowed to settle for 16 hours. That is very, very important. 16 hours you also stored, allowed for settling. Okay, at the time only 2 layers present. Next one. So I have also tried about screening all the seaweed. After that, Cypnia, Entomorpha and Sargassum. So we are also taking only for biodiesel from this particular seaweed only. Okay, next. So after storage, you are also stored in that container, very good container plastics. After that only you are also characterization, analysis of all the characters. Okay, next one. So nowadays, all the member now we are also going for Jatropha and other plants. Now in India also now started the biodiesel from marine algae. But foreign it is already going, now USA also going a large scale production in biodiesel. Okay, next one. Now basic biodiesel. So I will show some important slides. Okay, next one. So world biodiesel production 91 to 2005, how much increased? Easily to understood that emerging market already that 2005 the biodiesel also production also increased, day by year wise increased. Okay, next one. Here, that 2011 to 20, again the bioethanol production and biodiesel production, that biodiesel production only very high, millions of liters. That's the latest report. The biodiesel increased day by day. That's only told another 10 years, fossil fuel is a question mark. All the members mainly used for biodiesel, no doubt. Okay, next one. So US biodiesel, renewable diesel market, there is a millions of gallons, mainly the sources of EPA. That's only that mainly that biodiesel, mainly that good demand and marketing production also very high. Okay, next one. So conclusion, already the biofuel products produced from renewable biomass such as marine macroalgae and microalgae, the sustainable energy source, the great potential for carbon dioxide neutral production. Already all of them were well known that all that plants also taking from carbon dioxide and release the oxygen generators. So, all the mainly conference, everything talking about global warming, how to reduce the carbon sources. Mainly that God creating everything. Suppose you also culture in seaweed. The seaweed also give that nutraceutical and other nutrients purpose. The oxygen also give to ours, human being, and taking all the carbon sources. That's only you are taking for different aspect, or nutraceutical aspect, food, fodder. This is one of the aspects of biodiesel production. Okay, so that is only the carbon dioxide is very, very important, and this will overcome the energy source, environmental need to integrate technology and more precise economic assessment. Energy is very, very important aspect. And biodiesel about more than 80% energy density, kerosene, but can solidify the low temperature, high altitude flight. So that's very, very important. Micro and macroalgae is the only source for biodiesel. Okay, next one. So now all the countries mainly used for ship or plate or train, everything mainly used for that only biodiesel from marine algae, no doubt. Okay. Next. So this is our team. Once again, thank you very much for giving me the opportunity to
Thank you, Dr.
chairman and
Ramanathan.
all other Eastern sir, all other algal specialists here, Gurunathan, Veera Gurunathan and Dr. Ganeshan, my good friend. Both of them are studying in the same time in that PhD in our CS in marine biology under the guidance of Professor L. Kannan and Rangasamy sir also here at Nagaraj. All the members, my close friend, good friend. I am learning from Avana, A to B, everything learning from viewers, seniors. So once again, thank you very much for giving me the opportunity. Thank you for your kind wishes and blessings. And Arun Kumar also here. All the members, my good friend. Okay. Once again, this is a very, very important conference. I appreciate. Why? This conference only invites all the self-help groups. That people only culture in the seaweed in our coastal area. That people, the backbone. Okay. So that's only we are also appreciate. All the members also coming here and attend the conference. Only this seaweed promoting, that's only the members, the family members. Okay. Once again, thank you very much for giving me the opportunity. Iswaran sir, Surya Narayanan sir, all the members, Yelumalai, my good friend, all the members, my good friend. Once again, thank you very much for.
Thank you, Dr. Anandaraman, for the nice, excellent talk. So, Dr. Anandaraman is available here, and anyone wanted to interact with him, he is most welcome. Very cordial person. Thank you.
Thank you very much. Tea time also discuss. Okay, no time. Okay, thank you.
Thank you very much. Thank you very much, sir, for the wonderful presentation. Now I request Mr. Bharadaraja, Assistant Section Officer, Mars Mandapam, to honor Professor Anantharaman, Dean CS Marine Biology, Annamalai University.
So we are having another renowned speaker with us. Next is Dr. Nivedita Sahu. She is a principal scientist of the CSIR Indian Institute of Chemical Technology, Hyderabad. And she is also part of CSMCRI. She educated from CSMCRI from South. She was she worked in CSMCRI for quite some time. So I invite Dr. Nivedita Sahu to deliver a talk on algal research, a legacy from Mars Mandapam. Thank you.
Good afternoon, sir, director sirs, and all audience. To my of course, I will just keep on saying, as my talk is on algal research, a legacy for Mars Mandapam. Why a legacy from Mars Mandapam? Just a brief, because this is the first place where I got my posting as a scientist at Mars. Then at CSMCRI Bhavnagar, and then deputed at Mandapam, where I worked for 6 years. So through my talk, I will be sharing my experience, whatever I had at Mandapam, and how I carried this experience for my future ongoing activities. So of course, here I will be showing, I wanted to show one technique I had introduced, that is grafting in seaweeds, using Capaphycus alvarezii of 3 different strains actually. That was brown, red and green different strains. That was my first work with former in charge Dr. Ishwaran and Ganeshan, that we have published also that paper. And we found that how the grafting technique, which can be also successful in marine seaweeds, and it was having very prominent type of the biomass also. It was quite different than the control plants. Next, please. So this is the things which I was showing. The statistical analysis also shows, and the publications supported this work. Next, please. Following to this activity, chemical characterization of this particular that also showed that different type of biomass having high yield, and this was also published in Carbohydrate Polymers in Webeck in 2011. Next, please. Of course, that I left. Then this is another things which I worked on a little. I'm giving brief on epiphytic association on seaweeds, which has showed many type of epiphytic associations like acidians and many others. And this was one of them, which was identified as a Sigmodia pumila, which has such a drastic way it has affected that it was their Red Sea like that we identify, and overnight a big mass of cultivation, specifically in Erwadi, we lost a mass of Geladiella acerosa. Next, please. This was also published, that was in current science. And carrying this epiphytic association in seaweeds, my work at CSIR IACT, where we have isolated several bacterial, and that was for symbiotic type of studies, where we found that one of the isolates which was giving a very interesting and important pigment that was identified and characterized as the melanin. Next, please. The bacteria was identified as a Pseudomosa, and this is the pigment which I am showing. It was identified with different chemical characterizations and other parameters as a melanin pigment. This also got published. Next, please. And this was with large-scale cultivation in Gracilaria varicosa, which was done from tissue culture in a small of explants in the lab and sent to land, that is in sea, in a box method which we was done, and this work also got published in 2016. Next, please. Going, as we are saying that how seaweed is also working in other applications, this is in wastewater treatment, where it was the Gracilaria varicosa things which was grown in brackish water in that time, and after 65 days of that brackish water was having other pollutant contents like high COD, and all 65 days COD load of that water was also reduced, and along with that co-culture was done with tiger shrimps. That shrimps were also found in high biomass of around 60 to 86 kgs per piece. Next, please. Certifying to another seaweed, this application, this was actually in an experiment which we have done in Gracilaria particularly, where we have extracted the things for polysaccharide, and after the extract, the pulp was mixed with soil in different concentrations, and in various crop plants in the spots we have done. Among this, the brinjal plant was also shown, and this is some of the data we got in different days, where it was having very good results in brinjal, both fruit and the plants, both as a type of growth, plant leaves, and all other parameters. Next, please. This is one more work which is showing from Mars Mandapam to IICT, where the Gratilopia, another one red seaweed, which was collected from Mars during my work at Mandapam. This was characterization and all study I did at IICT, Gratilopia filiciona. The polysaccharide which was isolated, it was characterized as lambda type of carrageenan, and it was also certified with various different characterization studies like FTIR and all other analytical tools. It also got published in 2014. Next, please. As we know that the algae can be used for the food applications, and starting from many type of applications, we all from soup to many other food products. So carrying this type of applications, I also extracted one agar-agar from the Gracilaria verrucosa, which I had seen in the last cultivation in C. From that, the agar was extracted, which was made as a dessert. And this dessert was distributed, next please, I will be showing, this was distributed to our dignitaries. It is showing that DST secretary and former directors of IACT Indian conference, the dessert was quite appreciated, which is, I showed that sweet dish from Gracilaria verrucosa. Next please. So with this, we have, with this continuing this activity, we found that the trading of seaweed can be also possible in Indian market, and we found that FSSAI, that Food Safety Standard Regulation of India, now they are also giving many type of ISO certificates, standard certification for seaweed products. I'll be showing in next please. These are the categorization code based on different type of parameters what they give for different vegetables. Now they are also including seaweed in that particular categories, like frozen, fresh seaweeds, then normal unfresh dried seaweeds, treated seaweeds, canned processed seaweeds, frozen seaweeds, like vegetables are given. So these all products are available. They should have to follow particular standard protocols which FSSAI used to regulate. All has to pass through FSSAI, particularly in food industries, for coming to the trade. So now seaweed can be seen in Indian trade with having FSSAI certifications. Next please. So these are another parameters, and we have also made on monograph in this particularly where seaweed has been included with the passing. These are the different type of all other algae and other all parameters with ISO versions, and the references I have given, and this is the recent amendments made in the FSSAI regulation which is there in this. Next please. This shows this how potentials now we all are also now accepting that can be we can, like in other countries, the seaweed can be taken as food. Now we are also taking. So it is already taking a value, and we are. It is also have been used in our day-to-day activities. Now jumping to microalgae, similar to seaweeds, these have also having applications in various human mankind's, starting from feed, food, bioenergy, biofertilizer, waste treatment, wastewater treatment, carbon dioxide captures, and many other applications. Next, please. So these are the some, again now we have started in some of the microalgae at IACT, isolation of different strains for various other applications and other activities. Next, please. In one activities, we were studying the hydrogen productions by having hydrogenase enzyme, where we have taken some reference studies of one particular species of microalgae known Chlamydomonas reinhardti, and we have compared with this. Next please, I will not go all details. This is the all detailed characterization of that particular studies, where we have found that Chlamydomonas vulgaris was having higher hydrogenous production than other things. In these studies, we took model as Chlamydomonas with reinhardti strain, and this was also published in 2-3 times in different reference. One of the reference has been shown here. Next please. Going to a wastewater treatments, here I am applying that algae in different bioreactors. So the bioreactors are actually, we are integrating with membrane-based systems, where we are using algal strains for treatment of different type of wastewater. That could be municipality, then domestic also, and some industrial, which includes dairy industries and one coffee industries also. All these were treated with different type of strains of algal species. Next please. So in this we have reduced high COD loads, BOD loads also, and in some cases we try to decrease the order and the color of the wastewater also. In another experiment, this is the co-cultures where we were doing that algal bacteriological symbiotic relationship, and it is also applied in co-culture. Next, please, I'll be hurrying in that. These are some other species with different biochemical activity. Please, next. Next. This is the facility right now I have created at IISCT for wastewater treatment. Here algal species we are integrating with membrane systems for, as I said, different type of wastewater from different sources. Main target is to reduce the COD and all other parameters also. And this water, whatever we are treating, that water we are reusing that for different purpose like washing, gardening and all. Next please. The facility which is there, we are showing to last time former DC he visited and all other dignitaries at IISCT. Next please. Again next. This is the pilot scale. So assessment, overall assessment, how it is better than the conventional methods. Operating cost is low, and of course the sludge whatever is formed in this algae-based treatment compared to sludge, it can be used with other valorized products. And overall energy, if you think it is quite economical and cost-effective process than the conventional methods, eco-friendly, and the water whatever can be reused for applications. It can be recycled. Another one here, one more we found, this is actually algae, whether it is present in air. Yes, it was also found in a study. We found that some samples collected during monsoon's time, then suddenly we found that was having algal samples. The spores were, maybe that could be spores which we could not further continued, but this work we had just started. It was so appreciated and we could able to publish. It shows that algal presence in air is very, we should be very cautious. It is allergen to health. It can affect like the persons who are having asthmatic and all, like health. So health type of things, this algal present in air can be very bad to those type of patients. So those type of study is very useful, and it can go for a long time, but it has to be continued. Next please. Finally, just whether algae has any in future, I concluded yes, algal trade, that means a way to Atma Bharat. And of course, for this, many other things which I have noted that here already lot of noted and high experts are here, and they will also support me. Yes, algal trade is actually a way to Atma Bharat. So Jai Hind. Thank you very much.
Thank you very much, Nivedita, for timely completing the talk. So she's again available for interaction. Any of the scientists or industrialists or the students can interact with Dr. Nivedita. Thank you. Thank you very much.
Thank you, ma'am. Please stay here for half minute. Ma'am, please stay here. Now I request my colleague, Dr. V. Veeragurunathan, Principal Scientist, to honor Dr. Nivedita Sahu, Principal Scientist, IICT Hyderabad. Now I request Dr. Mautsi to honor Professor K. Padmakumar, sir. Due to shortage of time, now we are going to international presentations. We will start the session in couple of minutes. I request Pramod Bhai to pin Professor A.T. Charle through MS Teams. And also I request Dr. D. Ganesh, Professor D. Ganesh to chair the session in person. Pramod Bhai, can you please find the international speaker? Please ping him on the screen. Respected Dr. Alan Critchley, can you hear me, please?
Yes, I can hear you. Can you hear me okay?
Yes. We are glad to have you here, and I would like to request you to
You can hear
introduce
me
Dr.
okay?
Yeah, we can hear you, please.
Hello?
Yeah,
Good.
I kindly request you
Greetings from
to
France.
introduce Dr.
Hello.
Radhika. Thank you.
Hello, everybody. It gives me great celebrating Dr. Ganesan's superannuation. And I've been listening to some of the sessions earlier, and I think that India has clearly an important role to play in the future of seaweed cultivation. So it's a great pleasure to talk about here advances in seaweed cultivation. And if I'm right, I believe that our speaker is from Korea, Dr. Mikyeong Clary Kim, and she's CEO of Ecophycotech from the Republic of Korea, to talk on the present and future of Korea's algae research and industry for inner beauty in the face of a pandemic. And I hand it over to you.
Good evening.
Hello.
Good evening.
Hello, Dr. Ranika.
So
Yes, we can hear you.
Now,
Yeah, I request
start?
you to kindly share the slides.
Yes, please. Yes, please.
Is it okay?
Please go to full screen.
Pramod bhai, unpin.
Yeah, it's good now. Please
Good.
start.
So,
Yes, thank you.
so good evening, everybody. So I'm very happy to contact on, online, in international conference on applied psychology. Yeah. So, thank you so much for your production. And now, before talking, so I would like to congratulate on the honor retirement for Dr. Ganesan, and I appreciate also to Dr. Ganesan and other staff members, as well as Dr. Dinoshi Kumar, who was a postdoc researcher in my companies, and for inviting me as a speaker on ICAP 2022. Why is the ecophycotech? It means the phyco is from the phycology. So at the first times, R&D based company, so we started, but now gradually we apply the phycology and macro algae. It means seaweed plus micro algae. So finally, we manufacture our product. Ecophycotech has a unique ability to secure the domestic microalgae species and to apply them to final product. And now we sell in the world the brand of Dr. MCK algae-based cosmetics. It's chlorella-based. This is spirulina. This is laminaria. It's from Fucoidan. So today I would like to say is how to recover the post-pandemic by using algae. So the famous books already said, global pandemics remakes history, as you know. So now, because of the pandemics, we act overall, So because of air travel is increasing day by day, and climate change, and destructions of wildlife habitat, and loss of biodiversity, and population density in city, and endless war, extra. So to recover this crisis, we need some, very standard global ecosystem. And also, we have to reduce carbon dioxide from the fossil fuel, and commercializations of clean energies we urgently need. So because of fossil fuels, the vicious cycle is keep going. So we need, to prevent a pandemic disease. Of course, we need a vaccine, but so we need also immunity for our health condition. So to reduce carbon dioxide, carbon footprint also, and we need also environment conservation. So we have to recover for self dependence to find alternative to prevent the next pandemic. So immune system is very important. Of course, another journal expert said, algae becomes the most promising and innovate raw materials for functional products. So already. So even fat decomposition's effect. So extra is very important area. So today's I don't need to say the experience data, but just Korea is R&D and industrial trend. Generally, I speak about it. So in the world, of course, in company management also, ESG is very important to value change, environment, social, government. So why the importance of algae rising in an era of post-pandemic disease? As have you heard already, inner beauty? So why inner beauty? So inner plus beauty is a combined word of inner plus beauty. It is not just to contemporarily improve the outer skin through artificial methods such as cosmetic. It means you take care of your health and skin itself beautifully and healthy through eating and lifestyle change habit also. So I'm sure the algae is the best raw materials for inner beauty. So because of excellent ingredient in functional food. So I explain, the prominent product from algae classifies on the basis of their market value. The high values include nutraceutical, for example, astaxanthin, and cosmetic and pharmaceutical. And the medium and high value is spirulina, chlorella, alga, alginate, carrageenan, etc. And low to medium value is fertilizer and animal food. And now bioremediation is very important. Now currently CO2 capture is the best important things for environment. So as you know, so the algae market size in the world, for example, is gradually increase. So by annual growth, average growth is more 6.5% for algae protein. As macroalgae-based product market also increased 5% annual growth, average growth gradually. So, for example, the hot issue is astaxanthin from Haematococcus grupialis. So, it's very important for pharmacological function and cosmetic function. So about R&D and industrial trends of RG in Korea, I would like to say here is located Korea, South Korea is next to Japan. Is the three countries, Korea, China, and Japan, account for 84% of the world seaweed-related patents. Of course, this data was 10 years ago, is old data, but it's possible to estimate, the three countries, the high percent of R&D. So this is Korea. This is Jeollanam-do and Wando Island. Island Wando is the 91% is produced in this area. So Wando is a very special area for seaweed also. So our Korean Society of Phycology is held, for Wando International Seaweed Symposiums regularly. So briefly, in 2020, Korea is the third largest producer of seaweed in the world, and is 1.77 million tons seaweed annually. So the almost is Saccharina, is Pyropia, and Undaria is almost 28% of total. In 2021, we export the seaweed. Is the best one is Pyropia to countries around the world, such as Southeast Asia and Europe, generating $619 million worth of profit. So as you see in graph now is Pyropia. This area is gradually the export quantity is increased. This is another, seaweed. So our companies, we already have attended on the project, huge research project, biodiesel from marine microalgae by Ministry of Ocean and Fishery. It's for 10 years. It's a huge project. It was a huge project, about $14 million fund. So we finished well. So at that time, Dr. Dinoshi Kuma had attended on our project. So this project, finally, we got very good result. In 2015, Marquard's biodiesel branded used diesel for automobile, successful in driving from Seoul to Busan. So with Dr. Dinoshi Kuma, we in the first 5 years we published a lot of paper. So we tetracelmis is micro green energy. We try to extract oil, but now the next research step is we'll apply the tetracelmis for raw material for cosmetic. So I said already, the Korean Society of Phycology since 1986. So since 2013, we published the algae. This is International Journal, CSIR. So I said already, Wando International Seaweed. So the title was Healthier Future with Algae. So it's more 200 persons attended already, yeah, and 10 plus 10 countries and 120 paper was presentation were done. So briefly, I see what kind of research in Korea now is about so Eclania cava for bioactive substance or the Pyrophia cultivation. And for virus SARS for activity, bioactivity from polysaccharide, and COVID-19 from seaweed antiviral potentials they studied already. And because of CO2 on carbon dioxide now, so we pay attention a lot to climate change also. So next, Korea huge research project in the future. So we prepare already. So by Korea Ministry of Ocean and Fisheries, and there is recycling of by so products for sustainable resource circulation, and the seaweed based by healthcare project we prepare, and East Sea Blue Carbon Center construction project for blue carbon sink in the east coast marine, and alternative seaweed meat. So for vegan, vegetarian people, so we now prepare a huge project also. So for the future R&D and industry, we need also alternative to mass productions of stable and sustainable and high efficiency biomass and extractor for future algae industry. So successful industrial conditions for sustainable algae is, I think, very important thing is how can the biomass of algae be secured stably. And next, it is necessary to secure algae raw materials or effective extract with a high international price competitiveness. And in the past, as a company policies centered on R&D, but in the future, collaboration is required by setting hub or agency in Korea for mass culture of overseas algae with a price competitiveness and effective functionality with the international abroad, institute or company. So in Korea, in post-pandemic areas, interest in personal health care has increased. So the demand for vegan products, I said already, vegan food, vegan cosmetic also. So based on natural ingredients such as algae is increasing. As you know, Korea is well known for the K-beauty wave, and there are more than 15,000 cosmetic manufacturing companies in Korea. They are interested in new natural materials, and the demand for cosmetics raw materials from algae is increasing. So in conclusion, increasing industrial demand for algae in preparations for a pandemic. It is necessary to foster clean energy, food, and beauty industry by securing facility for mass production of algae. And Alga Biomass Center in Gyeongbuk is a local government Korea. So we need a joint research for future food industry of algae among overseas. We have to exchange and secure some manpower among the overseas algal researchers. So we need a collaborator to required by Herb and agency in Korea for algal mass culture of overseas. Yeah, that's all.
Thank you very much, Dr. Kim, for a fascinating oversight. Can I just ask one quick question at this point, sir? In terms of developing the Korean industry, are you concerned over the limited number of species? And is that why you're looking for overseas collaboration to perhaps bring in new cultivars and types of algae to grow?
Because, our country is a very small country. So we have every time. So we need we have some limit of the quantity. So is the production cost is very higher than overload of sea. So we need contacts. We need a collaborator. So. For example, so we have to raw material to very big company. So the price is very important.
Mm-hmm.
But in Korea, product is a high price. So so we need now. So we are searching is a high competitive price and high efficient. Yeah.
Thank you very much. I'm looking to see in the chat. I don't see any questions. Is there anyone from the floor who would like to ask Dr. Kim one quick question? If not, then Dr. Kim, thank you so much for joining us, and particularly in your time zone. It's been a fascinating update, and please stay with us for the rest of the session if you can.
Yeah, thank you so much. Yeah.
Thank you very much. Over to the organizers now, and I saw Ian Nish pop up, but I'm not seeing the screen from the conference. Ian, are you ready to rock and roll?
As long as you're ready to show my video.
Okay, well, let's, in the interest of time, and I can see you online, if the organizers have got your video to roll, let's go over to Captain Seaweed to introduce himself. Ian, I think, is well known in the region to everyone, but Ian, please just introduce your topic, and then I hope that the video can be played thereafter. Thank you.
Okay, well, my video is about my 22-year journey from Mandapam Camp to Bali, where I live. So my presentation is divided into 3 parts. The first part is about the 4 years that I was involved, CSMCRI and Mars and PepsiCo in Tamil Nadu, during which time I worked most closely with Dr. Ganesan, who I believe is speaking after me. So you'll see something of what I was up to with Dr. Ganesan from year 2000 till 2004. The next section just shows what's happening with C6 Energy, Indonesia. C6, as you know, is represented at the meeting that you're all attending. And so I just put in a video clip, mostly taken by my colleague, Gigi Komodo, he calls himself. just showing what the C6 decombine technology farms look like. And the final third of my approximately 12-minute video explains two initiatives that I'm particularly interested in beyond the core business of C6, but including issues of data acquisition and sustainable development that provide an important context for C6 operations. Last but not least, I participated in this, well, partly because of my connection to my old friend, Dr. Gunnison. We've been 22 years since we first met. So it's kind of a homage to him and the other scientists who I've worked with in India.
Thanks, Ian. So could we please roll the video for Dr. Nish? I can see online Professor Ariel Kushmaro is also online. And I think, Ian, after you, there's Professor Kushmaro and also Anisha Hurtado. So we'll line those up as well. And then I'm not sure whether Dr. Ganesan actually finishes this session or whether we go back to the other session 2, seaweed products. Would the organisers step in now and roll the video, please? And then also, I think my
a pleasant day to
presenter as well here, Dr.
First
Ganesh,
of all, let me thank the
he can
organizers
take
for the
over
invite to
after
join
you
this
finished,
webinar.
Ian.
The current
So
status of the seaweed
I can see the
industry
screen
of
with
India
three of
is
us
in dire
on.
need of good quality
Is the video,
seedlings.
there goes the video. Oh, that's Dr. Hurtado's video. We need Dr. Nish's, please. That was sorry, it's my fault for going out of order, but I saw Ian online. I didn't see anybody else, and so I grabbed the opportunity to introduce Ian. Almost ready?
Yeah, thank you.
Yeah, technology is a wonderful thing. I mean, look, this is fascinating. There's no way we could have done this years ago, right? I'm in France, Ian, you're in Bali, Dr. Kim, you're in Korea, and the rest of you are in India. I think that we're, Professor Kushmaro is in Israel.
Thank you.
We're all over the world, and this is a wonderful thing to be able to do.
Well, as I show in my video, I used to go in Mandapam camp in the year 2001 and 2 to a little cyber cafe with goats and cattle wandering around, kind of under a tree at the refugee camp. And I would connect to the internet through a 300 to 600 BPS dial-up connection. And with a little luck, I could receive and send a few emails.
Yeah, I remember that. Ding, ding, ding. They called it the handshake in those days, remember?
Oh, yeah. Sometimes the handshake didn't last too long. But...
No, it didn't, did it? No. So what we have now is 100 times better. Okay, I can see in my top left-hand screen the main stage, and I'm hoping that they... Otherwise, may I suggest that since you had Dr. Anisha Hurtado's video, you roll that and then we come back to Ian.
Hello, sir. Hello.
Hello.
Sir,
Hello.
we don't have video of Dr. Yanish. So.
Oh, okay. So
Okay.
please show Dr. Hurtado, and Ian, can you email your video quickly to Vera?
Yeah, please.
It's on YouTube. I sent the whole connection to the organizer.
Oh, it's on YouTube. Okay, the connection to Ian's video is on YouTube.
Okay.
But while you search for that, maybe because you had Dr. Hurtado's presentation, I think, as a file, please show that one. Or we could go directly to Professor Kushmaro in Israel.
Correct.
But I think while you sort that out,
Yeah.
which one should we do?
Well, we'll do that, Kushmaro. That.
Well, I sent the link long ago. I got an email 2 days ago saying that they had the link and they were ready to rock and roll.
Okay.
Nelson is there. Nelson Bhattacharya is connecting with me by telegram. So I'll give the link to him so he can pass it on. In case
Okay.
it's been lost.
Professor Alan, we will go to video which is available. Dr. Anisias, because Professor Ariel is in the class. He will be back at 4:40.
Okay, so let me quickly introduce Dr. Anicia Hurtado from the Philippines. It is a tremendous pleasure. She and I have worked for a long time together since I worked in the Carrageen industry at what was then Degussa. And so I think she does have some very interesting things to tell you about increasing production of Capophycus seedlings for large-scale cultivation. So if you could roll the video, please, and then I would say if Dr. Ganesh is then available, he could take over for completing Professor Kushmaro and also Ian Nish's presentations. Does that work? Thank you.
Yeah, dear participants, right now the presentation of Dr. Anicia is available through the video, and we are going to see the video, and she will be available for the question session. Thank you.
Okay, and I just sent the link to Nelson Bhattacharya too. Should be in the room.
A pleasant day to everybody.
Okay, thank you. Thanks,
First
then.
of all,
I think we can
let me thank the organizers
just proceed with Dr. Nisha's presentation. Much appreciated.
to join this webinar. The current status of the seaweed industry of India is in dire need of good quality seedlings.
Yeah, sorry, I'm not
their
getting any
production.
sound at this end. It's
Hence,
very sort of
I purposely
stuttery.
chose this topic to share with the audience our experiences
Well, the
with fervent
technology
hope
still
that
has
this
a
will
few
help
issues.
the industry. Join me then as we navigate this presentation.
These things just go to prove to us that when things work well, they work very, very
To
well.
begin
There we go.
with,
We're on the second
let's
slide,
take
but
a
I still
look at
can't
the
hear
world cultivation
Dr.
production
Anicia.
of
Is
Capophycus and
there an
Yukima
issue
in
over the sound?
2019.
And
After mid-2000, Indonesia became the world's
Ian, you're
leading
still there,
producer
right?
of Capophycus.
Yeah,
Yukima, after
Okay,
it had
so the sound,
overtaken
the sound's working with
Philippines, which
the
pioneered
presenters.
cultivation
Just am
since
not hearing the
1970. The topographical
the
location of
track
the Southeast Asian countries,
of
namely
Dr.
Indonesia,
Hurtado's
Philippines,
presentation.
Malaysia, Cambodia, Vietnam, Myanmar, and Timor-Leste, favors robust and sustainable production of Capophycus.
Okay, maybe let's go back to Dr.
Since
Kim,
India
and maybe
and Sri
if
Lanka
you
are
ask you
still nascent
another
in
question,
eucalyptoid
anyone from the
cultivation,
floor,
it is expected that their productions need to
could I ask you,
increase.
do you have any
Oceania,
issues with
Latin America,
introducing
and the Caribbean
non-native
region
species for
contribute minor
cultivation
volume to the
in
world
Hello,
production.
Dr. Alan, you please mute
Yeah?
your mic. Your sound is interrupting.
Yeah. Sorry. Okay.
Thank you.
Any industry for that matter faces challenges, and seaweed industry is not exception. A pleasant day to everybody. First of all, let me thank the organizers for the invite to join this webinar. The current status of the seaweed industry of India is in dire need of good quality seedlings to upscale their production. Hence, I purposely chose this topic to share with the audience our experiences with fervent hope that this will help the industry. Join me then as we navigate this presentation. To begin with, let's take a look at the world cultivation production of Capophycus and Eucuma in 2019. After mid-2000, Indonesia became the world leading producer of Capophycus and Eucuma after it had overtaken the Philippines, which pioneered cultivation since 1970. The topographical location of the Southeast Asian countries, namely Indonesia, Philippines, Malaysia, Cambodia, Vietnam, Myanmar, and Timor-Leste, favors robust and sustainable production of Capophycus and Eucuma. Since India and Sri Lanka are still nascent in eucumatoid cultivation, it is expected that their productions need to be increased. The Oceania, Latin America, and the Caribbean regions contribute minor volume to the world production. Any industry, for that matter, faces challenges, and seaweed industry is not exempted. These challenges are the major ones. 1. Loss of biomass due to ice disease, epiphytic filamentous algae or EFA, grazing, leading to shortage of good quality cultivars or strains. 2. Lack of regulatory mechanisms, especially in the translocation of seedlings locally and internationally. Policies are still wanting to the present. And 3, there is inequity of income among the players of the seaweed industry, especially the farmers. If there are challenges, there must be solutions, and here we recommend the following. 1. Promote conservation of wild populations to preserve genetic diversity and possibly source out new cultivars or strains. 2. Strengthen infrastructure.
Hello, Dr. Anisha is not there. Situally,
My first time to Manapam camp was February 2000. I was there with a boatload of people from Mars. And we'd survived the Y2K bug. I'd been hired by Jennifer Tien from Mars to work with CMCRI Mars and PepsiCo. This was my first time to India. I learned that people had been harvesting Gelidiella around this area for many years, as seen here in these ladies in 1987. And again, I was seeing harvesters around there in July later that year. The PepsiCo activities were happening around Crusadai Island at that time. There I saw some really big cottony with a rather distinctive morphology that I've never seen anywhere else before or since. This is what the Mandakam camp facility of CSMCRI looked like in those days. And on the right you see where I used to do my emails. A few kilometers away I'd walk up there and sit at a terminal, and cows and the goats were around for company. I had the pleasure of working with Dr. Ganesan from my start of working around there in year 2000. Here we are out in the field, and here we are visiting his family a couple of years later. They were very gracious to me, and I'll never forget the time I spent with the Ganesan family. Here we are on the sea, and on the left we see a very young Dr. Shanmugam, just beginning his work with PepsiCo. Again, the boatload of bosses from Mars on the upper right, and ladies planting seaweed as they sit on the seashore. Anybody who knows me knows that I'm a boat man, and I saw a lot of interesting boats around this area, and I spent quite a bit of time under the blistering hot sun in some of these boats. I saw some interesting seaweed too. I saw some good, some bad, and some ugly. Anyway, by 2003, production was in full swing. PepsiCo was producing dry, then baled seaweeds by 2003. Then alkali-treated and biostimulant products started to be made by Aqua Agri not long after that. Eventually, by 2010, they were joined by other Indian enterprises, including C6 Energy, where I work now. Mainly, I work at PT C6 Energy Indonesia, where we process and farm seaweeds. So we'll let you have a look at that going on. Just sit back and watch as we let the good times roll. At the end of the day, the seaweed's been harvested and it heads off to the factory. So here I'll give you a quick look at the factory that the C6 has in Northern Bali. C6 farming and factory operations are developing nicely in the hands of the young. So should this old veteran of the seaweed wars join the throngs of boomer expat retirees, pickling his brain and idling life away? No, not entirely. I'm focusing on two issues. These are key to the long-term well-being of seaweed farming, but are not core to the immediate push into large-scale company farms. Namely, getting affordable data, hitherto unavailable, and meeting sustainable development goals. This provides an enabling environment for thriving seaweed farms into the future. The vision? Well, we need an evidence base evolving and effectively utilized, ultimately getting affordable, available, evidence-based biomass solutions. We need ecosystem services to be understood and well managed, resulting in humanity living in harmony with nature. We need ecoscape communities thriving in ecological economies. Poverty alleviated, then prosperity built. Finally, we need ecoscapes thriving under integrated coastal area management so that we have sustainable community development. Solutions from information technologies were in their infancy in Y2K at the little cyber cafe near Mandapam Camp. You could get a dial-up connection at about 600 BPS on a normal day, sometimes not even that. Nowadays, information technology, Internet of Things solutions are proliferating and reducing in price. Increasingly affordable, timely, and accurate data are increasingly becoming achievable. Plus, there's an overwhelming mass of proposed solution frameworks out there so that we can develop collaborations that bring scientific evidence-based solutions to tropical seashores. So, in the big picture, we have to ask the question, is life on Earth a system of ecosystem services provided just to benefit humanity? Well, for humanity, the practical answer to this question is yes. And our role is as stewards of the biomass systems that enable our existence. Phyconomists focus especially on the crucial services enabled by photosynthesis. We concentrate on coastal communities where land and water meet in ecoscapes that are central to human life. We blend sustainable development goals into feasible initiatives, ultimately aiming to achieve the United Nations Sustainable Development Goals in coastal communities. As seaweed people, we can develop an obsessive monocropping mindset, but we must integrate into coastal area life. Seaweed farms in integrated coastal area management involve a number of interest groups, including aquaculture, fishing and stewardship managers, government, NGO and private enterprises, tourism, recreational, transport and shipping interests, and there will be data swap transactions among intelligence gatherers, kind of a data economy developing. One of the main uses for all of this information is having the ability to track climate change and blue carbon efforts. We speak so much of climate change, but so little of it is being actually measured. Secondly, we can produce bulletins, predictions, and warnings for people who make their living in, on, and around the sea, including, of course, seaweed farmers. We will have multispectral and visible light biomass logging that will make it much easier to keep track of the health of our seaweed crops on farms. Also, mosaic mapping and collaboration tracking that is aided by remote sensing, such as by satellites and drones. Finally, we will have spatial data for plans and operations as integrated coastal area management is undertaken. So please join us in our quest to acquire affordable marine intelligence and get it to seaweed people and others who need it, and to use the data to achieve sustainable development with seaweed as a key biomass contributor. Thanks for watching.
Yeah, I'm not sure if you can hear me now, but I can't hear you, so I think if Dr. Ganesh could take over, that would be perfect.
I've had no video, no audio for some time. I just got it back when you started talking, Al.
Yeah, I just got it back too. Ah, great.
Well, that's why I make my videos, the soundtrack all has subtitles.
Well, I think the thing for the audience is that it is searchable on YouTube if they wish to, if other people didn't hear the soundtrack. But you're very good at making pictures audible, so it's very easy to follow even without the soundtrack, but it's improved with your voiceover. If I can just have a little punt here, and if Dr. Ganesan is listening, by the way, I'd like to speak to you about a review of Gilidiella for Journal of Applied Phycology. So I'm just putting in that little put there. And if Dr. Ganesh is there, I think he should wrap up and invite the next speaker. Is Professor Kusmaro back from his lecture now? Not hearing many responses. And you said I switched to headphones just to make sure it wasn't the microphone on my machine.
Hello.
Hello. Hi. Yeah, ask questions of Ian right now. And then anybody have questions for Ian?
Please ask the questions.
Audience, if you have any questions, please. If no more questions, we will go for other session. I am handovering the mic to Dr. Ganeshan, sir.
Thank you
Dr.
very
Alan, thank you so much. I think there are 3 lectures from CSMCRI. Bhavnagar is there. We will back around 4:45 for these lectures. For who is already there in that Kushama is there in the class. We will be back on 4:45. So we will be coming back on 4:45. By the time we will be having that lectures of CSMCRI scientist. Okay.
Thank you very much.
dear participants, so we are again have two more speakers in this session too. The first speaker is Dr. Arup Ghosh. Let me have a small introduction about the presenter. Dr. Arup Ghosh is a principal scientist in CSIR Central Salt and Marine Chemical Research Institute, Bhavnagar. And he is basically involved in most of the academic and scientific and innovative research program throughout his career. And he was one of the key persons to involve the agro techniques and the organic farming and seaweed-based biostimulant and all those things. And to his credential, he has been awarded the Best Technology Transfer Award in the year 1918, and this is one of the significant contribution made by him. And he has published many research papers at the national and international level. Dr. Ghosh has co-convened in many national and international conferences, evidencing his experience in the field of research. And he is one of the key scientists working in the area of algal biotechnology. And now I invite Dr. Arup Ghosh to make a presentation. Thank you.
Thank you, sir. Very good afternoon to all of you. I have been given only 10 minutes because of the late schedule. We are running a bit late. So I'll have to hurry it up. So let me start. I would be presenting mostly about the science of seaweed-based biostimulants. Next, please. Okay, so you can see that there are so many companies selling biostimulants, and most of them are actually imported. And main species, you can see, they are Ascophyllum nodosum, Macrocystis, Eclonia, so on and so forth. But they are very limited number of species, so they are basically growing in cold countries. But if you compare those from India, you can see that there are predominantly few species that can yield biostimulant. Among them, Capophycus alvarezi is the most important one, followed by Gracilaria species and Sargassum species. And there are many entrepreneurs, industrialists who are already doing these products. For example, AquaAgri, Snap Alginate, C6 Energy, Excel Industries, they are already into these. And there are several others which are coming up. They have licensed technologies from Indian researchers. Next, please. Yeah. So how do the different biostimulant properties fare among each other? You can see this AquaSap. This is actually Capophycus alvarezi, and our licensee MS AquaAgri, they conducted a detailed analysis of what are the amino acid constituents. And you can see that Ascophyllum and AquaSap, that is Capophycus, they are fairly similar to each other, and in many aspects they are better than even the imported species. So to make a point that the indigenous, that is the species growing in Indian conditions, they are also equally having good potential. This is also going to cater to Atmanirbhar Bharat, but at the same time you also get a quality product. So be it Sargassum or it is Ascophyllum, of course the constitutions vary, but mostly they will contain this indole acetic acid as growth hormones. Other than that, cytokinins, and there are very many quaternary ammonium compounds that are giving stress, that are like stress busters like glycine butane and choline chloride. Other than that, few of the species are potassium accumulating. So in spite of being in seawater, they actually accumulate potassium. So you can also derive potassium from the rhodophytes. These are the red species. Next, please. Yeah. So what are the other constituents? Most of the constituents which we know are basically the growth hormones. But other than that, there are several other bioactive constituents which we try to analyze. We have analyzed several of them, but there are several other which we are still in the process of discovering. For example, you can see that the seaweed species, they contain a lot of polyphenols, and polyphenols are actually very good for imparting stress tolerance and other beneficial characteristics. Even there are very many species of polyphenols as such also, and that also we have characterized. So in spite of having different composition, the efficacy for both the cases, we can see them as positive. Next, please. Yeah. So CSMCRI, we have several technologies depending on what sort of water we are dealing with, the coast condition, the tidal wave condition. This is example in Tamil Nadu where we use the raft method. Next, please. And here in Gujarat condition, you can see that a lot of biomass has been harvested. And we here use tube net method because the tidal waves amplitude are fairly high. So the problem is, do we have sufficient feedstock? And I think the government of India is right now stressing very much on making or ensuring sufficient feedstock availability. But once sufficient feedstock availability is there, the technologies, downstream technologies are definitely there. Next, please. This is an example of I think Aqua Agri Processing Private Limited, they started a decade back, but now, they have set up a big enterprise, and this is these pictures pertain to their industry, which they have set up it in Manamadurai. So the basis of, the success are also due to a lot of trials, validation trials that has been carried out over 20 states of India, including 9 to 10 crops, even more. And by collaborating with at least 43 agricultural institutes all over the country, totaling up to more than 300 trials. Right now, the recommendation for FCO is 3 trials only at 3 different agroclimatic conditions, but we have validation trials for more than 300 trials. So that's the success of Kappa ficus, and every place we saw that about 11 to 34% of yield improvement has been noticed over and above the recommended dose of chemical fertilizers. So all these reports are available. Next, please. These are the universities which we tied up and did a lot of trials. Next, please. This is again the trial reports. I'm not going into detail, but you can see that rice, maize, green gram, black gram, sugarcane, potato. So many crops, all carried out at more than 20 places, 30 places, like that. Next, please. We also have found about 25% decrease in chemical fertilizer dose is possible by use of these biostimulants. So we recommend the integrated nutrient management, at least reducing 25% dose of chemical fertilizers. Next, please. Yeah, we have done several other research, even, when are the stages that are most appropriate when this seaweed biostimulant work. And that is very important for economic purpose also, because if you apply seaweed sap at a stage where it is not required or it is not responsive, you're simply wasting your money. So depending on the right stages, we have identified, and of course a lot of studies have to be done. Next, please. We have done detailed work on photosynthetic efficiency, how the photosynthesis improves, how the electron transport chain is enhanced in the chloroplast membrane, then water efficiency, water use efficiency, and other parameters were also improved. Next, please. If somebody asks about how these biostimulants actually work in plants, you can see it works both under stress condition as well as normally irrigated condition. See, the internal environment of the plants are modulated by modulation of indolacetic acid, kinetin, zeatin, salicylic acid, and abscisic acid in the plants itself. So these biostimulants actually does something that stimulates the internal composition changes. Next, please. Yeah. So I will not go into too much detail, but this is a slide wherein whatever we have found, we have actually published. So these papers can be read for all the analysis. So every point has got one paper at least attached to it. So if somebody wants, they can have a glimpse of these papers. For example, how sustainable this yield improvement can be. It is in cereals or sugarcane, pulses, oilseeds. We have recorded improvement in yields due to increased root length and root volumes. Then identification of best responsive stage also was done. Then deciphering the bioactive compounds and mode of action is something that is of interest right now also to us. And we have deciphered the role of auxins, gibberellins, cytokinins, and quaternary ammonium compounds. Even about the beneficial effect of these biostimulants on soil also we have done. And we have found improved enzymatic activity and nutrient transformation, higher nutrient uptake rate, soil microbial population also detailed study we have done. Next, please. Yeah. So even under stress condition, the uptake of nitrogen and iron elements, they improved by application of seaweed biostimulants. How that is done? Next, please. So we tested it on all the parts of the plants, whether it is seed, stem, leaf, root. So and how it is done? Next, please. Yeah, how it is done, we have elaborately tested by way of doing transcriptome analysis, wherein this mRNA sequencing has been done. And next, please. Next. Yeah, so what are the physiological changes that happens? What are the gene expression, which genes gets differentially regulated? That also we have studied both in leaves as well as roots. So I'm not going to go into the detail, but I mean the papers can always be read. These are in public domain also. Next, please. This is differential expression of various transcription factors. Transcription factors, you know, I mean, they are very important in regulating many other genes. Next, please. Yeah. So we have also found that how abiotic and biotic stress also can be warded off by use of seaweed biostimulants. Next, please. In that case, it was a tomato fungus called Macrophomina fasulina. So this particular example is how seaweed biostimulants actually improves the number of certain culturable bacteria like Bacillus subtilis, Bacillus licheniformis, Pseudomonas. So these are very important for nutrient transformation in the soil. Next, please. Yeah, this also maintains the diversity. So next, please. I'm not going to explain in great detail because we have a shortage of time. But anyway, I mean, the normal irrigation condition, there is a certain diversity and balance of bacteria, which under stress condition, if you apply with water, this balance gets disturbed, which gets restored if you actually use these biostimulants. And this is another example what we have studied by metagenomics, so high-end sequencing. Next, please. Same thing, how different taxa of the bacteria gets emphasized. Next, please. Yeah, so I'm not going, again, several bacteria get specifically improved. Next. Next. Yeah, not only for Carpophycus, we also have several results for other seaweeds also. Like, for example, like I told for Carpophycus alvarezi, we did 300 trials. You can see the same number of trials were also done for biostimulant derived from Gracilaria edulis. So similar, I mean, multi-locational, multi-crop trials were conducted all over India. For Sargassum also, that is a brown seaweed, but that's not cultivated, that's a collected seaweed. We have initiated several trials in fields also. Next, please. Next. Yeah. So I think I'm towards the end of my presentation. One important thing is that now biostimulants fall under FCO category. So these are the specifications that have been mentioned. you have to have certain minimum, restrictions by way of content of the heavy metals. And at the same time, you have to at least do about 9 toxicity studies, many of them acute and some of them are there. So these are the things which one has to do if somebody has to actually commercialize them, the formulation. Next, please. Yeah. Also about climate change impacts, we have also found that this is having a very low carbon footprint. We have analyzed with the life cycle assessment impact software, that is GABI. And definitely we have a good climate change impact. The carbon dioxide equivalent emission per unit of production, it gets decreased by use. Next, please. And that's all. That's all in short what we at CSMCRI do on seaweed biostimulants. Thank you very much.
Thank you, Dr. Ghosh, for nice presentation. I think due to the shortage of time, if you have any doubts personally, you can approach Dr. Ghosh and clear your doubts. So we will, now I request Dr. Satish to honor Dr. Arab Ghosh for his presentation. So now I am going to call upon Dr. Pushpshil Shukla. And again, he is from Central Salt and Marine Chemical Research Institute, Gujarat. And he basically worked on the understanding and mode of action of seaweed-based biostimulant in mitigating abiotic stresses and improved nutritional use efficiency. And he is currently working as scientist in C6 Energy Private Limited. And he is also involved in the next generation biostimulant for sustainable agriculture. So I would like to call Dr. Pushpshila Shukla for a presentation. Please, madam. Please limit your talk probably within 8 to 10 minutes. We are already running short of time.
Yeah, I will try. So thank you, Dr. Ganesh, for introducing me. And since I am an alumni of a CSMCRI, it's an honor to be here and present. Like it's getting a full circle. And so since last 7 years, I'm working on this. Last 7 years, I'm working on this biostimulants. So before, it's a brown seaweed, and then it is a Pramod bhai. Yeah. And then now it's working with the Kappa ficus-based seaweed. So today's topic of my talk is that seaweed-based biostimulant, whether it is causing any insulin-based approach to improve plant growth and stress tolerance. Why I choose the word insulin-based? Because agriculture in India is one of the most unpredictable phenomena. So we don't know what will happen to the plants when it is in the field. So using the biostimulant, whether they provide a kind of an insurance to the farmer, you use these biostimulants, and in the future, if something is coming to your crop, it will protect. So next, please. So this is basic things when the plant is growing into the field. It is facing salinity, drought, salt, and different pathogens. So if we use biostimulant, so they are inducing a kind of a natural immunity in the plant. So we call this an induced systemic tolerance. So if it has a tolerance against these abiotic stresses and induced systemic resistance if they have a resistance against pathogen. Next, please. So this is a generic mechanism by which plant offer a stress tolerance. So there are several signaling molecules that induce the expression of certain level of a gene. So when these signaling molecules will bind to the transcription factors, they regulate the downstream expression of the further genes and produces subsequent protein. But the production of this subsequent protein requires a huge amount of energy. So when we see that there is a stress tolerance in the plant, it is followed by a kind of a energy trade-off that leads to the change in the phenotype of the plant. So that is our objective, that if we use these biostimulants, whether we can minimize this energy trade-off during the stress tolerance. Next, please. So for that, we are working on the seaweed biostimulants. So as everyone knows, like they are all expertise in seaweed cultivation. So seaweeds, like they are the cellular organisms and basically classified in 3 parts depending upon their pigment: brown, red, and green. So in nearly throughout the world, there are 47 companies that are dealing with the production of seaweed-based like agri inputs. Next, please. Yeah, so this is like these seaweeds are rich in different bioactive components. And they have those bioactive components in the seaweed extract. They help plants to cope up with the different stressors in the future. And so again, I will emphasize while an insurance-based approach, because they will prevent an energy trade-off. Like if we are making a transgenics, we are inducing only one gene to promote any stress tolerance, and that will come with the energy expense. So again, so these seaweeds, like they contain, so normally I have made that figure with this alva and sargassum, ascophyllum, and kappa ficus. Those are the 4 examples for the different classes. And since ascophyllum and kappa ficus, they are the more general seaweed biostimulants, so I will take that as an example. Next. So if I say brown algal seaweed extract, so ascophyllum is most researched brown seaweed extract, and I worked on that seaweed from the last 5 years. And so they are rich in various bioactive compounds, like if we say phyllotannins, unique polysaccharides, amphicoidins, mannitol, and laminarin. So these are known to induce plant immunity and plant stress tolerance and confer like tolerance to the crop. So if I say other name of the brown algae, there are Eclonia maxima and laminaria and sargassum. They are also used for the production of biostimulant. But if I say like ascophyllum, throughout the world is the most commonly seaweed, brown seaweed that is used for the production of biostimulants. Next. This is the study that we published. So this is the expression profile of microRNAs. MicroRNAs acts one step down of the normal gene expression. So we see that if we treat the plants with the ascophyllum extract, they confer more of a salinity tolerance than the control plants. But we try to explain it on the basis of a molecular basis. So if we see that after 6 hours of treatment with this ascophyllum extract, we see the plants treated with the NE in presence of the salt, they behave as it is like control plant. But when we initiate that to 12 hours, there is a all red expression, that red, we can see in the salt that is completely red. So it's induced like all the stress tolerant genes are induced. But in the NE treated plant, the extent of that induction is less. Means plant is conferring the stress tolerance without causing an overexpression of the genes that is helping to cope with the different stresses. Next. So this is like on the basis of the different studies published. So we try to formulate a mechanism by which they are conferring the stress tolerance in plants. So I will not go in detail. So there is still a research lagging behind what are the receptors that they accept bioactive compounds from the NE. So in turn, they induces different mechanisms like carbohydrate metabolism, cell wall biosynthesis, secondary metabolites. And that help into the expression of the different genes. So next. And similarly, this brown seaweed-based extract mitigates the drought stress. So again, this is the study from on the glycine max. We can see in the figure, like before stress, they are normal. But when we provide this drought stress, and in the recovery phase, the plant treated with the asco extract, they recover better than the non-treated plants. So there is another interesting that the bioactivity of those ESCO extract that is basically dependent on the process that we are using to design a biostimulant. If we are using an alkaline hydrolysis, they have a different set of metabolism. If we are using an acidic analysis, they have a different mode of action. And if we are using a neutral ESCO, they have a different mode of action. So the amount of the bioactive compound produced mainly depend upon how the seaweeds are processed and what is the process of their development. Next. So, but there are few limitations with the brown seaweed. Limitation is mostly the brown seaweed is naturally harvested. They don't have a humongous cultivation system. And second, its bioactivity varies by different procedure, that extraction procedure. And third, from where this is harvested. Like if I have a escophyllum that is harvested from Scotland, and I have a escophyllum that is harvested from Maine, they both have a like entirely different biochemical composition. And the third, the season in which we harvested. So there is a huge variation among the different batches of the biostimulant produced using ESCO. The next. So then is the other example is the red waste, in which I am currently working on the C6. So basically, C6 works on the production of this biostimulant from Kappa ficus. As Sri mentioned in his lecture that we have revolutionized that cultivation in the large scale using a C combine. And these are the different commercial names like Agrogain, AG-Fort, and AG-Boost. So Agrogain is basically for plant growth and stress tolerance. If I say AG-Boost, it is for inducing the immunity in the plant. And AG-Fort is for immune induction. So in general, the carrageenans are present in Kappa ficus. That is known to be a bioactive component. Next. So in a paper, I have deciphered that how this carrageenan promotes this stress tolerance. So there are three kind of, so the interesting thing that we have a three carrageenan like iota, kappa, and lambda. These all three carrageenans have a different mode of action. So if it is an iota carrageenan, it will induce a different set of genes in the plant. If it is a kappa carrageenan, it will induce a different set of genes in the plant, and it helps the plants to counter. So the plants knows like what sort of chemical is sprayed onto the plants, and they will behave in response to that. So next, so Agrogain. So from now I will speak on the product that C6 made. So it's a natural biostimulant that has an active ingredient, and it has a defined mode of action. Next. So this is that characteristic why make Agrogain unique. Because it is scientifically proven, it has a consistent performance. And I will not go in detail in this slide. Next. So all the batches of the Agrogain that is passed through a strict quality control because they have a definite chemical signatures. So all the batches, if will be considered as a biostimulant only, they matches this chemical signature. And so all the time this Agrogain has the same specific bioactivity. There is no variation between batches to batches. It is tried throughout the world, having different trials in different parts of the world, increasing on the different crops. And this data is from the multi-season and multi-location trials. And it works on different crops equally well. Next. So these are the geographical locations where Agrogain has been sold, and it is shown that consistent performance throughout the world. Around 22 countries, 100 plus research trials, and overall 10% more than increase. Next. So this is the general, so those are the gene expression of the tomato plants sprayed with the Agrogain. And they induce a different set of the gene that is helping the plant to grow better when they are sprayed. This ESCO extract induces around, if I say more than 50 unique genes, that helps the plant to grow better. Next. So this is the graph. We can see this. There is a more of a root in the Arabidopsis in the Agrogain treated plants. Play the video, please. Video. not playing. Or you can skip. I can show this. Yeah. Okay. So this is that video actually I showed in. So this is a time-lapse video of rice seedlings grows in nitrogen. So we can see that last 3 chambers has an nitrogen and first 3 chambers has the control. We have a better grown rice seedlings in the agrogen plant as compared to the control. The next one. And the same thing is with the P. We have seen the better root system in the agrogen treated plants as compared to the water treated plants. Next. Yeah, so this is the clear indication, okay, if we treat this 0.1 gram, 0.1 ml per liter of an agrogen, you can see the third picture, the roots and shoot is much better as compared to the other sets. Next. So, and this is a stress tolerance. So what here comes this incidence-based approach. The foliar spray of the agrogen is improving the stress tolerance as compared to the control. So this is like if we sprayed the plant, and in the future salt stress is coming, the plant will, the crop will perform better as compared to control. The next one. Yeah. So I think it's just finishing this. Yeah. And this is just by reducing. We have seen that plant treated with the agrogen, that reduces ROS in the leaves. Yeah. Next. So the chemical fertilizers, we know there are different harmful aspects of the chemical fertilizers, and it reduces, like there is a over usage of a chemical. So if we can use this agrogen and reduce the input of a chemical fertilizer in the field, so this is the slide that shows how chemical fertilizers impact human life. Next one. So we can see if we having a different nitrate, known level of a nitrate in nitrogen deficiency in different nitrate condition, when we apply the agrogen, the plant performs better even in the nitrogen starved condition. But if we go to the full nitrate, there is no difference into the growth. Since this means that only in the starved condition, if we apply the agrogen, it helps in the more growth of nitrogen. Next, yeah. Next. And that too by changing the leaf architecture. So we can see the agrogen treated plant, the pattern in which the leaves are emerging is entirely different from the control under the nitrogen starved condition. This shows that they also have an impact on the developmental models. Next. So the same, that it reduces the ROS under the nitrogen starvation too. Next one. Yeah. So there is a definite pattern on the nitrogen starvation and the flowering. So we see if there is a high nitrogen, there is a less flowering. If we have a less nitrogen, there is a less late flowering. So we need an optimum level of nitrogen for inducing flowering. So next slide. So if in this slide, we can see that in agrogen treated plants, in the nitrogen starved, we have early induction of a flowering. So if we treat our crop with this agrogen into the nutrient deficient condition, it is inducing early flowering as compared to the controls. Next. Yeah, so this is the summary that how it is a solution for the sustainable agriculture. So I will not go in detail. And the next one. So this is a take-home message that using the seaweed biostimulant, are we trying to make a balance between the growth and the stress tolerance without reducing the energy trade-off? Yeah. And thanks. So next. And so these are the guys, my mentors who supported my research throughout my research career. So Dr. Balakrishnan Prithviraj, Alan, and Sri, he always supported my ideas, and Dr. Pradeep Kumar Agrawal being my PhD mentor, and all the colleagues of C6. Thank you very much. Yeah.
So thank you for the nice presentations. And now Dr. Sada.
I request my colleague Dr. Kanjan Trivedi, scientist CSMCRI, to honor.
Dear participant, I think now Professor Ariel Kushmarov from Israel, he is on the line and he is going to present his talk. And let me have a brief introduction about Dr. Ariel Kushmarov. So he is an Israel. He completed his postdoc in Harvard University, and he has made a tremendous career improvement in many universities, and he has made many publications. There are many honors. For example, Seisman Institute of Science Award. Then he has got a plenty of experience with many societies like Israel Society of Microbiology, then International Society for Microbial Ecology, and many professional membership he was. Now I would like to call upon Professor Ariel Kushmarov for the presentation. So please share your PowerPoint. Madam, can you please share your PowerPoints?
There is opening chat, but I can't share the presentation somehow. Just a second, I will send my.
Hello, sir. The season has ended up.
Hello, hello. Mic check. Hello. Check, check. Hello, hello. Hello, hello, hello. Hello. I have a small announcement for all the participants and delegates. Kindly have your tea from your sitting place. Thank you. Tea is arranged just behind the hall.
Can you please, all of you, assemble back? Professor Ariel Kusramov, am I audible?
Yes, I'm with you. I'm online.
Okay, you can start your presentation.
Thank you very much. So I'm sorry for the inconvenience and
Proceed with your presentation.
Yes, I'm starting. The topic of my presentation is complexes of copper polysaccharide from the red sea microalgae, and we will include characterization and bioactivities of those compounds. And co-author with me is Nofar Yehuda and Professor Shosharad, both of them expert in algae. Next slide, please. Someone has to change the slides. Thank you. Our institute is located in the south of Israel, and you can see the lab and the infrastructure. Next slide, please. Next slide. Slide number 3. Another slide, please. Okay. It's very important to understand the efficiency of bioconversion of solar energy by photosynthesis, and obviously later on to receive a byproduct or a product that we can have some beneficial or profit from it. Next slide. Here we can see several examples from Israel, open ponds for culturing different moieties from Dulaniela, and this is in the south of Israel, nearby the city of Elat. And the main production here is beta carotene, again using the power of the sun in order to do some beneficial products from algal culture. Here we see another plant in Kibbutz Torah, also in the south of Israel, and this time culturing and producing hexaxanthin, and this is on glass pipes, a very nice production and very accurate in terms of condition. Next slide, please. And here we see a culture of porphyridium, red algae, that isolated from the Mediterranean Sea. And this microalgae is growing in plastic tubes with aeration. And the main commercial product right now is sulfated polysaccharides that is very essential for the cosmetic industry. Next slide. This algae is unicellular, about 10 to 20 micron. Its habitat is could be brackish water, seawater, and fresh water, and several photosynthetic pigments, including chlorophyll. Next slide, please. What's important in this red algae, the porphyridium, is the outer polysaccharides that behave like a capsule to the algae and give some protection. Next slide, please. Here we can see the production of the polysaccharides, and we see a growth curve of the algae and polysaccharide production and viscosity. And what we see that some of the polysaccharides is bound to the cells, and low percentage is dissolved. And along the growth of the community, the viscosity is going up. And actually, in the process of production, we extracting mainly the dissolved polysaccharides for the media. Next slide, please. So this sulfated polysaccharides, as I mentioned before, is very durable and have a lot of interesting moieties, and its structure is actually looks like gel. It's built out of xylose, glucose, galactose, and glucuronic acid, and more other substances. We don't know exactly the structure of the polysaccharides, but we know that its molecular weight is quite high, between 3 to 5, 10 to the 6 Dalton. Next slide, please. So this is a little bit information about the structure of the polysaccharides. Next slide, please. These polysaccharides, we assume that it's actually provides some shelter and defense to the algae, and it's have antibacterial, antifungal, and antiviral activity. It's also behave as a buffer layers against extreme environment, absorbing light, change of pH, etc. So the sulfated polysaccharides till now can use as part of our aim in algal biotechnology, and can use as a product depending on the environmental condition, chemistry modification, etc., etc. Next slide, please. So the potential application of these sulfated polysaccharides is cosmetic and used in dermatology. It could be mixed as gelatin agent in food. The algae can also provide sulfactories. The activity of antiviral is very important. And of course, we have some experience that it could be used for joint lubrication, etc. Next slide, please. Also, the property that also make it important for cosmetic, it's anti-inflammatory, antioxidant, anti-aging, and more. Next slide, please. Next slide, please. Next slide. Okay, what I want to share, it's when we adding metal to this sulfated, mainly copper, but also zinc, we changing the structure and activity of the polysaccharides. For example, in the panel of electron microscopy, we can see that normal biofilm of Acetobacter baumannii and Pseudomonas aeruginosa on a glass surface, and below that it's if polysaccharides only cover the glass, and if we're using polysaccharides combined with copper, then we see that there is no biofilm, and a minute amount of cells are attached to the surface. Next slide, please. So here we see the antimicrobial activity of this complex if we adding zinc into the mixture, and actually we see its activity, and we see the inhibition ratio. So we see that depends, of course, on the concentration, and we see that if we have nice inhibition on gram positive and also gram negative bacteria. Next slide, please. Next slide. So actually what's happening, it's that we have a co-polymerization with the different metals, and now we'll concentrate it on copper. Next slide, please. Next slide. So our hypothesis of this research, it's that copolymeric copper with the polysaccharides are results in synergetic bioactivity against fungi and bacteria. Next slide. Next slide. Next slide. Yeah, okay. So this is the way that we cultivating the algae in the lab. This is in a room with plastic sleeves. and we using about 3% CO2, temperature of 25 Celsius, and we separating the soluble polysaccharides from the media, and then mixing these purified polysaccharides with different sorts of copper. Next slide. So part of our testing will be that I will show you its FTIR, surface morphology, AFM, and antimicrobial activities. Next slide. So what we see here, it's FTIR of the polysaccharide mixed with copper, and we see that only copper oxide is forming a bond or covalent bond with the polysaccharides, and actually it's very stable and there is no leaching out from this structure, even if you're using 500 ppm of copper. Next slide. What's interesting is the change of structure of the polysaccharides, and this is using AFM microscopy. And if we look at this panel of the polysaccharides alone, we see that the morphology is quite flat. The spike height, it's about 24 nanometer, and density about 500. If we have a mixture of copper sulfate with the polysaccharides, in this case we don't have a covalent bond, the heights of the spikes is about 20 micron and density of about 1000. And if we're looking at copper oxide, then the heights of the spikes is 1000 nanometer. And actually this is also important in the activity, and we see that the copper oxide polysaccharides actually is more effective in killing bacteria, and we see here example of E. coli and Bacillus subtilis. Next slide. And actually here, and this is, we think, very important, very interesting, we can see the activity against Candida albicans, that the copper oxide polysaccharides killing more than 90% of the Candida albicans in surface and cultures. So, and also when we check cell viability of the treatment, we see that most of the cells are dying. The next slide, yeah. We're asking a question, what is the mechanism and how it's working, and does there is oxygen stress or without oxygen, etc., etc. Let's see what's the answer. Next slide. We see that this kind of salt, copper salt that we're using, it's also changing viscosity, changing conductivity, and also the potential. Next slide. And this is again FTIR of the different salts, and we see that actually monovalent, like copper oxide and copper chloride, are forming a covalent bond in the polysaccharides according to the FTIR, and there is no leaching out. Next slide. Here we see again the structure, and actually when we have a copper oxide or copper chloride with the polysaccharides, we see that the height of the spikes is about 1000 nanometer and very high density between 2000 and 5000 spikes. And actually think about a surface with a lot of this kind of spikes, what it's doing to the bacteria or the fungus. Next slide. So here we see a summary of the inhibition, and actually we see that the copper monovalent, copper oxide and copper chloride are more active, and actually we think this is due to the height of the spikes and the sharpness of the spikes. Next slide. Also, it's influencing the spreading of bacteria. And when we're adding copper oxide or copper chloride, there is no spreading of bacteria on the plates, and compared to the other kind of salts that we're using and the control. Next slide. So the possibility of the mode of action is hiding in the spikes, and actually when we're looking at a spike sharpens aspected ratio, we can compare this kind to holes in the membrane, and we can use it by luminescence. I cannot go to the method here, and we see as the height of the spikes is going up and the sharpness is more effective, then we see more bioluminescence, and actually we think this is the mechanism of killing the microorganism by the polysaccharides originate from the red algae in this research. Next slide, please. So actually, if I summarize, we use algal culture in order to produce sulfated polysaccharides, and then down the line we mix this valuable product with copper salt, change the structure and change the activity, and now we have a new product with high durability as antifungal and antibacterial, and hopefully this new product will go into cosmetic and into clinical studies in the near future. And thank you very much to all of you.
Thank you, Professor Ariel Kushrama, for your detailed presentations. So, I think we are going to have the final presentation by Dr. M. Ganeshan, Senior Principal Scientist, CSMCRI Marine Algal Research Station, Mandapam.
Dr. Kamlesh sir, are you available on Teams?
Yes.
Sir, kindly share your slides.
Can you see?
Yeah, now I would like to introduce the speaker, Dr. Kamlesh Prasad. He is a Senior Principal Scientist, Central Salt and Marine Chemical Research Institute, Bhavnagar. And he is the most eminent scientist, received many awards, CSIR Young Scientist Award, then visiting professorship in Lyon, France, Fellow of Royal Science Society of Chemistry, London. So, and beside this, he has received many awards, and he has published many research papers, particularly in the field of the seaweed. And now I would like to call upon Dr. Kamlesh Prasad to make a very brief presentation, probably within 10 minutes, 8 to 10 minutes. Thank you.
Okay. Yeah. Thank you, Dr. Ganesh, for the introduction. And again, am I audible now?
Sir, I can't see your slides. Can you please reshare the slides again?
Just a minute. Can you see?
No, sir, it's...
Can you see the slide? Now?
Can you please share entire screen? Yeah, now it is visible. Yes.
Okay.
Thank you, sir.
I think when I'm sharing, I think I will present like this only. So I hope I'm audible to all of you.
Let's make it full
thank
screen,
you very
sir.
much. When I'm making it full, I think that time, is it okay? Okay.
Please go ahead, sir.
Yeah. So thank you very much for this. And before starting my presentation, I just wish a healthy and very happy post-retirement life to Dr. Ganesan, one of my colleagues, and done excellent work in the seaweed biology and especially in cultivation. And I'm associated with him for last 22 years. And I'm very happy to be part of this conference. And before to start my talk on the marine resources from valuable products from marine resources. So marine resources are divided mostly in three parts are there. So seaweeds, apart from seaweed, crustacean shells are there, and then the fishes. So there are three things are there which are main resources of marine resources. And so from them we can get nutraceuticals, antioxidants, polysaccharides, and functional fluids. And apart from this, the cosmetic uses and pigments and lipids and biofuels are also been used from marine resources. So these are some of the structure of some polymers, or you can say polysaccharides, which are derived from marine resources. For example, most of you know about agar, carrageenan, alginic acids, then alvan, fucodans, chitin. So this is chitin is from crustacean organisms, from shrimp shell, or you can get from prawn. And then limonarians are there, then chitosan. So from chitin you can get chitosan. And apart from polysaccharides or polymers, marine-based pigments are also very important. So they are coming from phycocyanobillins. You can say blue-green algae, red algae, or cryptomonas. So these are the things we can get phycocyanobillins. And phycoerythrobillins are also present in red seaweeds. And then the chlorophylls are there in green seaweed. Then fucoxanthin in brown seaweeds, then astaxanthin from crustacean organisms. So we have worked a little bit on all these species. And then since my talk is on neoteric solvent systems, it may be a little bit new for some of you, but the neoteric solvent systems are divided into four things, four classes of solvents are together known as neoteric solvent systems. So these neoteric solvent systems are consisting of ionic liquids, then they are consisting of deep eutectic solvents, then biomass derived solvents, then supercritical carbon dioxide. So these all four solvent systems are known as neoteric solvent systems, and then have been used and finding applications as a solvents or as a co-solvent in various pharmaceutical applications, in catalysis, and in biotechnology, in separation and purification, and as a functional materials, and electrochemistry, and in heat storage devices. So these are some of the application of neoteric solvent systems. So a little bit chemistry about solvent system is that you can see in the left hand side, this is the ionic liquids, And these ionic liquids, just like sodium chloride. Sodium chloride is a salt, and these are also salts. But the difference is that in salts like sodium chloride, sodium and chloride both are inorganic in nature, and they bond through ionic bond. So very strong bond. And to break sodium chloride, you need to heat it for 800-degree temperature. On the other hand, in this particular case, the cation part is an organic moiety, and the anion part can be an inorganic or organic moiety, and they bind through only electrostatic interactions, and that is why the lattice energy of these particular salts are very low, and therefore they remain liquid at room temperature. So this is the difference. And apart from this, depuritic solvents, so these are the solvent systems which we can prepare simply by mixing a hydrogen bond acceptor. For example, choline chloride, it is a hydrogen bond acceptor. It can accept hydrogen. And then hydrogen bond donors. So hydrogen bond donors can be any acids, can be any alcohols, can be any sugars. So they, when mix the hydrogen bond donor and hydrogen bond acceptor, and we heat it for some time, so we get a liquid which is called depuritic solvent. So apart from this, they are more greener than ion liquids. They're easy to prepare, less expensive, less toxic, more thermally stable. And then this bio-based solvent systems, I am giving the name of only two bio-based solvent systems. One is ethyl lactate, one is lipidic acid. So these deuteric solvent systems, proof of concept has been done, and it is evolving now. It is emerging field, and it has been used for the extraction of seaweed polysaccharides such as agar, carrageenan, alginates, even and from crucidian cells, chitin, then fucose, mannose, galactose. And this is normally also used for the extraction of fucoxanthin, astaxanthin type of natural pigments from seaweeds. And these particular products have been used for the formation of ionogels, aerogels, films, electrolytes, biocomposites. So even nanomaterials, even grafting, antibacterial polysaccharide. So a lot of applications have been found, but everything remains in the proof of concept level at this point of time. And in our laboratory at CSMCRI, we have done extraction of agarose using bio-based ionic liquids. So what we have done exactly, as you know, if you have worked with processing of seaweeds, so when you are someone want to extract agarose from agar, they have to purify. You have to remove sulfate from agar to get agarose. So that is a very lengthy process and very expensive process. So, and what we have done here, we have tried to add some ionic liquids. You can see bio-based ionic liquids on the right hand side of the table, having the structure like choline formate, choline acetate, choline caproate, choline capryllate, choline laurate. And we try to extract, coagulate the agar from this seaweed solution, seaweed extract directly at a room temperature without going through any purification, which conventionally done. So that is how we try to understand the mechanism, the chemistry of the extraction, and we can see the yields have increasing when the carbon chain length of the carbon is increasing from formate to laurate. So that is why we try to understand why it is happening. So we found that some chemistry behind it, and we found that whenever the Kraft point of the surfactant, because all these ionic liquids, when the carbon chain length is increasing, the side chain is increasing, it behaving like a surface active molecule, site like surfactants. So they have a Kraft point. So whenever the chain length is increasing, the hydrophobic chain length is increasing, the Kraft point is increasing, and it is increasing the solubility. So more the solubility, better is the precipitation of agarose due to the formation of choline agarose complex, because agarose normally when in the seaweed, so there is counterion by sodium and potassium, or whenever we are adding choline laurate or choline acrylate type of things, then this is precipitating choline as a choline agarose complexes. So then we have tried to understand the solubility of carrageenan also in various ionic liquids, and that is we found that some imidazolium based ionic liquid can solubilize carrageenan, not only kappa carrageenan but iota and lambda carrageenan also it can solubilize. And the reason behind is that our aim was to find some common solvent system for cellulose and carrageenan. Since both are soluble in different solvent systems, so we cannot make composites. So we found some ionic liquids which can solubilize both cellulose and carrageenan in the same ionic liquids. So that is why we are able to prepare some composite gels. gels. And this concept will later on we have transferred to the deputic solvents. So in deputic solvents, what we have done, we have used some hydrated deputic solvents to extract carrageenan from Copperficus alvarezi, and we have found that in presence of hydrated choline chloride glycerol, the yield increases to 60%. So it's a substantial increase in the by 25% increase in the yield was observed in when we compare with the conventional method. So this is a substantial increase, and we try to understand the chemistry behind. So why this particular and deputic solvents are helping in extracting in better way. So we have done some intermediate study that we have find out some which the structures we have confirmed by NMR studies, and then we have found a particular conditions when you are using some deputic solvents, for example, choline chloride, ethylene glycol, urea, and the sulfate content remaining intact. On the other hand, when the temperature of, is increasing or the pH is increasing, the sulfate content is reducing. So that is the reason why the increasing the yield of the carrageenan at particular hydrated ionic deputic solvent conditions. So again, we have also, this is the work where we have converted a toxic, so-called toxic ionic liquid to a non-toxic entity by converting it to a alginate derivative. So in this particular case, tetramethyl ammonium hydroxide, which is known to have some toxic effect. So what we have done that we have converted, we have extracted the alginate derivative of the salt, means tetramethyl ammonium alginate we have prepared, and we have checked the toxicity against some phytotoxicity we have done against some chlorella species, and then also we have checked its seed germination studies as well. So we have found that these particular derivatives which are prepared from a toxic ionic liquid, and the derivatives are non-toxic to the soil microbes and non-toxic to the chlorella also. And that phytotoxicity phytoremediation study also has been done using duckweed. So it helps that by conversion of a molecule by a seaweed polysaccharide can make it a little bit non-toxic for applications. So apart from this, we are also working on phycoerythrin, which is a red pigment present in the red seaweeds, and this particular pigment, our phycoerythrin, has a lot of application as an antioxidant, as an anti-tumoral, in cosmetics and medical diagnosis, pharmaceuticals, and food industry. And but this phycoerythrin is very expensive. It is sold in the market. Even sold in the market is very high cost. So, and reason behind is that the extraction method is very tedious for the phycoerythrin, and the yield of the product is very less. So then we started our research, and we have collected a lot of seaweeds from the west coast and southeast coast of India, and then we have also developed some good buffer ionic liquids, so of these structures, and then we try to extract our phycoerythrin using these particular good buffer ionic liquids. Then we found that certain good buffer ionic liquid can extract phycoerythrin without the contamination of phycocyanin. Now, normally what happens when you're extracting the pigment using a buffer solution, you are getting a contamination of phycocyanin with phycoerythrin, and the separation of phycocyanin and phycoerythrin is quite difficult, and that is why the cost of the product is very high. So we have developed a process. We have got about analytically pure purity index or analytical pure product with using a particular goods buffer ionic liquids, and the production cost therefore has reduced drastically, and now very lab scale production we can do at 1 gram at 10,000 Indian rupees.
Dr. Kamalesh, we are already running short of the time. Please.
Okay, just I'm listening.
Okay.
Yeah. So I think I'll end here because there are more slides are there. So just last slide, I'll talk about the challenges of nutrient solvents and the strengths. So nutrient solvent systems are recyclable, and efficient biomass configuration can be done, and it can provide stability, environment friendly, and safe. But at the same time, a lot of challenges are there with nutrient solvent system. For the scale up and the cost of production is a little bit higher. And also there is always a product of contamination issue is there when you are getting the end product. So that already these problems have to be solved using with time. So thank you very much for listening to me. Thank you.
Thank you, Dr. Kamalesh, for the nice presentation. Now this is the last presentation going to be presented by Dr. M. Ganesan, Senior Scientist.
Good evening to everybody. Respected Director of CSMCRI, respected representatives from different companies, universities, colleges, research institutions. I hope it is concluding speech of today's seminar. So I would like to highlight some of the achievements, important achievements I have made during my tenure in CSMCRI. So absolutely I said it is not possible to tell everything within a period of short of 30 minutes. I would like to highlight whichever is important to the audience, important to the CSMCRI as well as nation only. Next slide. So I joined as a junior technical assistant in CSMCRI in 1992. So before that, I was doing a PhD in Annamalai University, Parangapettai, under Professor Kannan, who has recently expired last month. So he was not happy that when I showed my interview card to him. So I have been called for the interview in CSMCRI Bhavnagar for junior technical assistant post. He said, I am against your appearing interview because you are a highly qualified person writing your thesis PhD. You have already published 2 international papers with a high impact factor and high citation. So you would be welcome for the scientist post, not for the JTL. I said I cannot claim immediately, catch that top branches of the tree. I have to hold that lowest branches and then climb step by step. So he said, anyhow, you go. And then when I went to the Bhavnagar, and I don't know that anybody there in 1992, I think it was there in the February. So I was sitting in the interview hall. There are 3 insiders who are already, I think they were doing their PhD and then postdoctorate there in Bhavnagar itself. They also appeared, and I have decided that this post will not be for me. Some other that local persons, that insiders. So I got very much upset and went inside with courage that absolutely this post was not there for me. And I was really fought with the interview committee members when they said your PhD data are not correct. I said that I showed my earlier published reprint. This is correct. This is absolutely possible in Tuticorin coast. Then said it's okay, you go ahead. And then I presented well for 20 minutes and came out. And automatically I was unexpectedly, I was selected for that post. So I joined here as a junior technical assistant at Mandapam. Immediately I was assigned to study that iodine-rich seaweeds. So I made that survey of entire islands, I think for 2 years, the 20 islands beyond the depth of 20 meters, and found that plenty of resources of Asperopsis taxiformis, which has 0.5% dry weight of its body is having iodine content. And another important seaweed is Padaina, which is abundant in the coastal region. It is having 0.3% of its body weight is having iodine content. So I successfully tried and then successfully cultivated Padaina gymnospora, that is Moyajesni, by spore producing method in Yerwadi as well as Kursada Island, and produced good amount of biomass in a unit area. But I could not try that deep water Asperopsis, which is now emerging a lot of interest to study that iodine content, which is successfully done in Australia. Next one, please. And another one important seaweed which I have cultivated is Hypnea. So Hypnea musiformis is an indigenous carrageenan yielding alga. So it is very, very simple method which I cultivated by that Koiro in Kurzade as well as Irwadi. where I got that it is a 15 days of the growth of 250 gram to 300 gram per meter square I have obtained. If you multiply in a huge area, we can have that tons of Hypnea musiformis can be cultivated successfully over here. So at present junction, we are very much, I think, positive of seed material for Kappa-ficus alga. So this is the excellent alternative source for the carrageenan and also that's also here at indigenous alga. So which is now, whatever work we have done at CSMCRI by me and other scientists are now emerging interest of that nation, emerging interest of all other scientists. Next one, please. So this is another important alga. Just now also we have discussed with our director in the restaurant that high media is terribly wanted alga with which is giving that bacteriological grade agar. So agarose is one of the alga which is available in Gujarat, having very good agarose Gracilaria dura. I suggested that Gracilaria Gelidella acidosa is one of the best alga. It's giving that bacteriological grade agar, so which is highly marketable, highly valuable. And another thing is it is very, very difficult alga to cultivate. So I have tried several methods. That's coral stone method, that's 4 different methods, that raft method and cement stone method and net methods. So for 2 years we have very difficult this alga to cultivate. We have, I think, a lot of difficulties. We have successfully came out that this methodology, cement stone methodology is working well for the cultivation of Gelidella acidosa at commercial scale. It was published in a very good journal and we got very good biomass also from that compared with coral stone. Next one, even we have not stopped with that. We wanted to develop another best method which is easier method which can be done by the farmers easily. So we have developed a method called hanging stone method. So it has been a very small stone we can tie in the a raft in the rope, and after 120 days we get very good tremendous biomass which is superior than earlier methods which we have tried. And our snap alginate Dr. Negimiasa visited this farm site and at Irwadi, and he has immediately said that I will be taking this Gelidella cultivation technology. So this is the wonderful alga. I hope that even after 10-15 years it would be coming into the limelight. So this alga would be, it's most essential. It should be conditionally cultivated for getting bacteriological grade agar. Next one, please. So this is another, what we have done some through tissue culture method and developed strains which are far superior. And having, you see that agar content is around 1,500 gram gel strength, which meets excellently the quality of bacteriological grade agar. So this was published in algal research. Immediately, this is the first paper from CSMCRI published in algal research from the seaweed point of view. Next one, please. And this is another alga. That's my predecessor has cultivated Gracilaria edulis, Dr. Thomas, Dr. Subbarao Maya, everybody done. They are doing that, they did but rope method and that long line method, which they faced severe grazing. So I have adopted this method very fast, that raft method of Gracilaria edulis cultivation, which is being cultivated in all places, Thoniture as well as there, which is, I think, very promising technology of, I think, even 1.5 kilo of seed material is giving, I think, 25 to 30 kilo of biomass. It is a very promising technique. Even today also we are adopting this type of technology. Next one, please. So this is another interesting alga I have tried and got succeeded is Gracilaria salicoria. Nowadays it has been abundantly available along the Park Bay coast, and all our industries are now harvesting it for agar as well as for extraction of biostimulant. So we have successfully cultivated this alga for 2 years completely, and every year we got 5 harvest average, and maximum biomass we got it 60 kg. We have just tried with a little different method, that's a tube method in a raft. It's worked well, particularly for this alga. And this is also, you see that agar content is around 450, which is highly suitable for food grade agar. And all our industries, agar industries located around Madurai, are harvesting in bulk quantity, almost 400 tons per year. They are harvesting from the Park Bay coast. And all other industries are also harvesting this for extracting sap. You see that table having that Gracilaria salicornia, having a lot of ingredients of potassiums, hormones, and everything. Almost that it is equal to the Kappa ficus. This is another wonderful alga that we have developed cultivation technology. Next one, please. And this is also an interesting alga, which is also called Sarconema filiforme. Very, very rare species. This is also indigenous species giving the carrageenan. So you see that right picture shows that the carrageenan content, it's a hybrid of lambda and iota carrageenan. It's not a kappa ficus, kappa carrageenan. It's a hybrid of lambda and iota carragenans. And it's also similar to, almost slightly better than kappa, so it is Hypnea. It is very soft and maximum of 4 to 4.5 kg only. We got that biomass from a meter square area. It's also a very good indigenous carragenophyte, which can be taken up by anybody at any time. Next one, please. And coming to that, we have also tried to go little offshore for the cultivation of economic important seaweeds like Kappa ficus as well as Gracilaria edulis. We got a project from MNRE to develop offshore cultivation technology for commercially valuable seaweeds. We went almost nearly 10 kilometers from the shore. Imagine that even during the rough season, we went and put up rafts over there after 10 kilometers where even the Gulf of Mannar you see and Park Bay very, very shallow. Even after traveling 10 kilometers from the shore, we got just only 5 to 6 meter depth. So we put up our rafts, and the rafts are growing very well, both the Kappa as well as Gracilaria edulis. We got comparatively and significantly higher biomass yield and DGR while compared with both algae in onshore region. But the only thing is we faced a lot of difficulties over there. So the main difficulties is fishing. Fishing is the very big problem. They are making the trawling nets 10 kilometers away from that Uchipuli, from that nearly that Kushi beach. Farmers at night, they put long 10 kilometers, they are trawling net. They have put it. What happened when next day when went there, all rafts are entangled with that fishing nets. That's the fishing is the very big problem in the Gulf of Mannar as well as Park Bay region as far as offshore cultivation is concerned. And another one major is Maritime Board getting permission. It's very, very difficult. We tried for 6 months and even could not succeed for getting permission in offshore cultivation of any alga, even indigenous alga. Next one, please. And this is another important R&D we have tried also and success. And this is the cultivation of edible alga, which is an emerging trend currently. And government is looking for cultivation of edible seaweeds in coming years. And we have experimentally, we have success in cultivation of this through spore techniques, which is that the first picture is vegetative parts. When it became matured, the thallus is became a yellowish color and a lot of germlings. What we did, we put on a stress condition, by put a whole day in the plant in the dark condition. Next day morning, we put it in the flask where thousands of spores have been released and attached over the rope. When it was taken into the field, it has grown well. So this is one of the alga which is we have tried, and the development of technology was also done. Next one. It's a similar kind of algae, the similar kind of experiment we have done in another alga of edible seaweed, that is Alva compressa. Next one, please. So coming to that, apart from the cultivation, we have surveyed that entire Gulf of Mannar stretch, starting from the 0.5 meter to up to 30 meter stretch, where until that which depth seaweeds are available. We have found excellent number of seaweeds. Our team has, with those days, 10 years ago, we got very good divers, permanent employees who can able to dive even 30 meters with snorkeling and diving equipment. And we have collected, next slide. So we have collected and recorded 282 species of seaweed diversity along the entire Gulf of Mannar coast, out of which 40 are new. And we have prepared an entire book. So the book has been accepted in 2020 by Cambridge Scholars Publishers. So due to COVID, the process was really very slow because most of the printers are not coming to the office. I think probably the book will be published in the month of June, maximum, or July 2022. And it will be a Bible. It's a very good field guide for the identification of students and researchers for the seaweeds in the Gulf, not only in the Gulf of Mannar, almost whole Indian coast. Next one. And these are also, I have worked on that DOD drugs project and sent the material, and we have promising activity for nearly 6-7 seaweed species tested by CDRI Lucknow. All those species are available, only collected from Gulf of Mannar only. Whatever the species available from Gujarat has not shown any promising activity. Next one. So this is another interesting, very recent work we have done is spore culture, commercial culture method of Grasslaria adulis. It's a very simple method, but it's innovative method where we can release the spores in the lab and culture over there for a short while in the lab and transfer to the, I think, to the tank. And then third phase will be the sea. So where the overall concept of this, this is a work is, we could able to get, I think, 1.5 kilo of biomass from simply 8 gram of Grasslaria adulis with reproductive part within the period of, I think, 3 to 4 months of the period. This is the various steps involving in the commercial production of Grasslaria adulis through spore. Next one. So this entire work was done in our recently built that seaweed seedling production facility at Mandapam, which was opened by our Director General in 2019 July. And this is you see that success of this story. 20 rafts at a time we have harvested and taken the photographs. After seeing the success, Dr. Radha Mohan Singh, that time that United Agricultural Minister has visited to Mandapam, and in Tonithurai, he has distributed this spore cultured Grasslaria adulis material to the farmers of Mandapam as well as Erwadi. Next one. So this is another method which is attractive nationally today, and CSMCRI has been, I think, speaking for developing this technology and developing this facility. And not only Tamil Nadu, the entire states, including Andaman, wanted to have this technology and this production and the facility all over India. So this is also a simple technology which has been, we got literature from, I think, Anisha, and we have completely changed according to Indian condition and the Indian condition of our own cup of acres plant. What is the similar way? The concept is we are getting from the 100 gram fresh weight of the plant, 300 seed material with elite quality. Elite quality in the sense higher biomass, higher DGR, as well as superior carrageenan content. So after seeing that initial success of this plant, this project, this is Giriraj Singh, Fisheries Minister, came to I think Mandapam and visited our CSMCRI facility and distributed the seed material to several fishermen. And next line, we have given these seedlings to all 3 districts of Tamil Nadu that where cultivation is going on. And we also studied that in our own experimental farm that the growth is superior, and our chemistry team has analyzed that carrageenan which is quite far, both in sense of yield as well as gel strength. Next one. So this is another important, nationally important project where I am working currently. That's cultivation of seaweeds in Andaman coast. So it's a very, very difficult project, and we have faced a lot more of difficulties in establishing farm in Andaman. It's not so easy, and ultimately we got success in establishing seaweed cultivation in 3 different places. One is South Andaman, Hathitapu, another one is Middle Andaman, and the third one is Diklipur in North Andaman. So I still remember morning when Sri Kumar has shown that slide close to the Indonesia, which is having very calm water. I said that just now before discussing here, coming over here, Maya Bandar is an excellent place in Andaman, very calm, even during the monsoon, even during the cyclone. And it's hundreds, I think more than 500, 600 hectares are there for the cultivation. So if that commercial scale of cultivation is established in Maya Bandar, it could easily feed the entire requirement of seaweed, the Kappa ficus, essential for our country. Next one. So this is now currently going on where seed material is being distributed through the, I think, seed production. Next one, please. So these are the different pictures which has been done by myself and my colleague. Next one, Dr. Tirupati. Next one. So this is the last month we have distributed the seedlings to the farmers by the district collector. Next one. So, and I have, with my curiosity, developed that several facility in Mars Mandapam. This is the one done by, with the funding support of national NFDB. This is having that very good area for the cultivation of edible seaweeds. Next one. This is a tissue culture facility I have done with the support of funding agency from NFDB. And we could able to produce 10,000 tissue culture micropropagules in the particular facility. So it's a wonderful facility which work is currently going on. Next one. And I think being a scientist in charge, it's during the very short period of 1 year, I could able to create some sort of infrastructure. I think with the valuable support of the director who is sitting here. Whatever I asked for the development of infrastructure at Mandapam, he never said no, even today also. And I have done that name board, as well as I have converted on 2 rooms. One is in that excellent seminar hall, and another one is excellent discussion hall, conference hall, where every VIP are coming and having the discussion with us. So this is a very short span of 1 year I could able to make with a valuable support from Director of CSMCRI. Next one. So these are all my guru and mentors and well-wisher. Without their support, they are supporting us throughout my education career and throughout my career in my CSMCRI. Professor Kannada is no more. He passed away last month. He was a very valuable professor. And Professor G. Subramanian was my teacher in BSc Majora College. And Dr. Pushpito Ghosh, who is the person identified me when I was a technical assistant. He said he is the person with the huge potential. He should be elevated as a scientist. And when he became the director, the first order he gave us, appointment order, he has given to me. And a lot of support I got from Dr. Amitava Das. And I think huge support I am getting from Dr. Kannan and Dr. CRK Reddy for the last 30 years. I think without any quarrel, any different of opinion, is my elder brother. And these 2 are industrialists who had a lot of affection on me. So after seeing my work, and Professor Rangasamy sir is it's like an advisor for me. Each and every step he will be interacting with me. And then also that Dr. Padma Kumar, so he was senior to me in CS in marine biology. Besides which I cannot get their photographs, or Dr. Kaliya Perumal, who was very first man helping me while I came to Mandapam for my PhD studies. He provided his house for my stay and supported me throughout my PhD study. And also two persons to my seniors are Dr. Ramarao and Dr. Subbaramaya. They are excellent supporters. So I always remember, even after my career, these 10, 12 persons who were my mentor, guru, and well-wishers and supporters. So next slide. With that, I think I take a bit from CSMCRI. And I always remember the service I rendered in the CSMCRI. I never forget whatever I have achieved, and not only me. So whatever we have achieved, scientists from CSMCRI 10 years, 20, 15 years ago, is now come into the limelight. Government is wanting, private industry is wanted, everybody is wanted that list. So CSMCRI, highly, it's a very, very prestigious institute of CSIR. It's coming on one of the topmost 5 institute in terms of external funded, in terms of patent released, in terms of technology released, in terms of I think paper publication. In all the sense, CSMCRI is topmost of top 5 institute. I always remember my work and my institute, parent institute CSMCRI in my lifetime, and all my supporters, well-wishers, which I have shown in the pictures, which were not in the pictures. I always remember them. Thank you very much, sir. Thank you very much. Thank you.
Now I thank Dr. M. Ganesan for wonderful, inspiring lecture about his research journey at CSMCRI. I request Dr. S. Kannan, Director CSIR CSMCRI, to honor our renowned scientist, Dr. M. Ganesan. Mr. Pramod, honoring on behalf of Cooperative Society, CSIR CSMCRI. I request all the staff from Mars Mandapam to assemble here on the stage. Dinesh, please.
என்னுடைய பிரதர், both of them same time working in the PhD under the guidance of Professor L. Kannan. ஸோ இந்த இணைய நிறைவு விழாவிலே பங்கேற்று அவரை கௌரவிப்பதில் ஐ ஆம் வெரி மச் ப்ரௌட் ஆஃப் டாக்டர் கணேசன், மை பிரதர். Now I invite our CSIR Director, Youth Director, Sir Kannan sir, please. Vachina honor review. இந்த ப்ரோக்ராமை சக்ஸஸ்ஃபுல்லாக நடத்தின இங்கே இருக்கின்ற அடுத்து என்னுடைய நண்பர் வீர குருநாதன் அவர்களை அழைக்கின்றேன். இவங்க தான் இந்த ப்ரோக்ராமுடைய சக்ஸஸ் டீம் பில்லர்ஸ் இல்லையா? அடுத்ததாக திருப்பதி அன்பு நண்பர். அடுத்த அன்பு நண்பர் சதீஷ். Once again, thank you very much. அடுத்ததாக நெக்ஸ்ட் we also expect our another seaweed conference. அது எங்க? You only decide. Okay, thank you very much.
So now we are almost in concluding session. I am happy to declare the best poster presentation award. I call one by one. There is no first and second, all are equal. For each session, we selected 2 best poster. So we have 4 sessions, so 8 poster, best poster presentation award going to declare. The first, Mr. M. Bharadaraja, CSIR CSMCRI. For seedling production for commercially important red algae Gracilaria deplies. Sir, please. Miss Rosini Prithviraj Swastika for catalyzing of seaweed epiphytic bacterial community. Next, Samadir Chandrani, MK University, for characterization of novel foliaretinine from Padina Detrostomatica. Next, Ganesh Vaishnavi, MK University, for the paper Exploration of Marine Brown Macroalgae. Please. Sir, Ganesan sir, please come to dais. N. Selva Sudha, Pondicherry University. Selva Sudha. For the work, development of seaweed polysaccharides. Dr. Kanjan Trivedi, for the poster titled Role of Copperbaigus salvaricis Seaweed Extract. Please come. Ms. Bayal, CSMCRI Bhavnagar. Bayal, for the work Division Pattern and Evaluation of Technical Performance. Ms. Danya, CSMCRI Mars Mandapam, for the work Elite Seedling Production of Copperbaigus salvaricis. So the posters are selected based on the judge evaluation, 2 judges evaluation. Congratulations to the winners. Thank you. So, I made my sincere thanks to our director for giving this opportunity as a convener to make a concluding remarks of this ICAP 2022. So, this conference was actually planned 2 months before to felicitate our eminent scientist, Dr. M. Ganesan sir. And within 2 months, is it possible to organize an international conference? We have that. Initially, we decided online national seminar, one-day national seminar. Later on, we have our newly joining colleagues. So their guide, one from Israel, one from South Korea, and our well-wisher, Ansi Guttado madam, and Jan Nies. So once it crossed 3 personalities from international level, it became an international conference. So we decided it is possible. So we declared as international conference. So what the title we should give while planning that? So it should be a general one, not restricted only on seaweeds. So with that international conference on applied phycology, because phycology, if we will deal with deal with algae, it is a classical one. But when it comes in the applied, so there are so many applications. So we formulated the title International Conference of Applied Phycology. And another thing, funding is the major one. So we approached first NABARD. So NABARD immediately they declared 1 lakh rupee for printing charges, souvenirs and brochures and all. They immediately sanctioned 1 lakh rupees sanction letter for this conference. Then NFDB and our own industrial collaborators, when we write a mail, I wrote a mail to Abhiram Seth sir and C6 Energy team, Dr. Nelson, and one more team, Snap Alginate. So Snap Alginate, we decided April 1st to hold this meeting, this conference. And April 2nd, we received 1 lakh confirmation mail from Snap Alginate, Dr. Naimia sir. Thank you, sir. Thank you very much. And the next day, then we get another sanction letter from NABARD. Then we get C6 Energy confirmation, and AquaAgri also we received confirmation letter. So we planned 4 lakhs rupees budget, but we get it more than 6 lakhs later from industry itself. So that is the greater enthusiasm we got it whenever we need some help, especially monetary benefits or technology and all.
Small announcements, all the participants are doing.
Okay. So immediately we got a help. So now next thing we need to get invitees. So whenever we have our KCP project, our institute things and all, so we invited 10 lectures for a special chief guest. So all are unanimously agreed. Within 1 or 2 days, they confirmed and they provide title. So within time period, this conference made it, and we made a circular by speed post. Wherever we posted, we got it minimum 2 posters from the respective college or university or academy. So today also we got it 35 spot registration. That means it reached in mass audience. So that much reply, we got it, huge response, we got it. So this international conference mainly dealt with the 4 important things. Only is what is the issue in seaweed farming. Second, how it can be implemented throughout the India at pan-India scale. So, and third one, what is the novel products? Generally, phycocolloids is the main product. So, what is the other novel product we can develop? That this conference also focused on that. And what is the novel extraction procedures? That also Kamlesh sir explained well. So these conferences made a clear-cut platform. Now, under PMMI scheme, there is a huge opportunity for seaweed farming activity with the subsidy and all. So we have a target from Government of India for each state. So we are moving towards this. The first step, we need to identify suitable site for implementation in other maritime states. We are traveling towards the goal, a big achievement goal. So we have our internal project also, same thing. We need to identify suitable site in all maritime sites. So in this, where the conference today morning, especially Commissioner Sir talk, have very good information, and also NABARD officials who participate here and sitting here, they made it very clear statement that NABARD is always supporting, including infrastructure facility and all with subsidy. So this conference made a very good platform for a new entrepreneur and as well as DIAS to exchange the ideas. So this one-day program may be fruitful and give a clear idea how to proceed further. In this, I request Ganesan sir to give final concluding remarks on this conference.
So, good evening to everybody. So, you are all in the mood of leaving to your respective place because time is almost 6:30. I would like to concluding this symposium by thanking all the supporters because I think this is the only physical mode of seminar after the pandemic period of 2020, March 24th, where that everything was stopped throughout the nation. After 2 years, this is the only physical mode of conference for phycology, algae. So everybody wanted to have a chat with this and had a discussion regarding industries, regarding that they want to listen that what the speakers are talking about, that seaweed cultivation as well as downstream processing. And in that sense, it is really, the conference is great success. With more than 100 people are sitting over here, and then more than 300 people are participating through online. We have very heartfelt, extending our heartfelt thanks to all the participants, besides who are the chief guests assembled here morning. And I think on behalf of our director, I would like to extend our sincere gratitude to, I think, three most important industrialists from India who crossed almost 75 years. They took a lot of effort from travel from their own places and reach here and sit from morning to evening. So I would like to mention number one, that Mr. Abhiram Seth, that Managing Director of Aquagri, and Mr. Vinod Negimia Sarab, from Managing Director of Snap Alginate Industries, and also, most importantly, Mr. Ashwin Shroff from Mumbai, who has generously accepted my mail and came here. And above all, our thanks are also due to Dr. Kannan, our beloved director, who is really that brain behind this organizing this conference. He wanted this conference has to be done in an excellent manner. Like this is the first conference after COVID pandemic. He has extended all his support, and he traveled all the way. Yesterday night he came to Ahmedabad around 12 o'clock and got up at 3 o'clock and catch the flight from Ahmedabad to Mumbai and Mumbai to Madurai. Such a lot of difficulties he made because of making this conference success. So overall, we thank each and every participant and delegates of this conference. And with this, I think few words, I would like to conclude this conference. Sir, thank you.
Thank you very much, sir. Finally, we have one small announcement. As per scholars' request and staff request, all CSMCRI staff would like to have a photography with Ganeshan sir, followed by we can take a photograph of all the participants who received the presentation awards, and we can have entire team photography also. Thank you very much. I request all the staff from Bhavnagar too, students, staff, everyone, please. Let's take a group photo first. Let's take a group photo first because some dignitaries wants to leave. I request everyone to gather here for a group photo. Participants, please bring your shield and certificate. I have another announcement for the Participants, we will send the certificate through post. And we will send the receipt also, payment receipt. Thank you for your cooperation. Right side we have lot of space. We will conclude the program with national anthem. Please 1 minute. We will conclude the program with national anthem. We will conclude the program with national anthem and everybody can leave. Please stand up. Please sing chorously.